So, very good morning to everyone. How are all of you? I hope doing well. Am I audible? Everything all right? Visual? Yeah. Audio, video, everything fine? Very good morning, very good morning, very good morning. So, once you inquire in all studio, it is coming properly or not? Ask another campus also, here also. Okay. Clear. Right. Very good morning. See, so the agenda of the today class is to revise fastly about the hits area, important area. So we are not going to teach right now. So simply I am going to say you what specifically from which line usually, from which chapter, which paragraph, from which line there is a chances of question is more. So just we will talk on that particular area in somehow in an emphasized manner. We are not going to teach the entire chapter. We are not going to deal the concept behind it. Simply we are going to talk about the very hits area in the last moment. Ki if any line match in the examination day, you just remember sir told in marathon class regarding KG, you just keep in mind and these are the things happen like this. It's all right. And definitely we will just move according to from where there is more chances of asking the question. So I'm not going to move according to the sequence of the NCERT chapter. We are just going to deal according to the weightage of that particular chapter. It's not a fixed weightage is there, but if we talk about the trend, according to the recent trends of 3-4 years, what are the more important chapter from where questions is going to ask more, we will just cover the more. Right? Done? Clear? So let's start. Definitely we are not going to waste more of our time. So let's start. So we are starting with the first chapter of the biotechnology. So what to keep in biotechnology in our mind? What are the area we have to work in biotechnology? So one thing you just, principle and process of biotechnology we are going to deal. First, restriction enzyme. Write the name. HIND2, don't forget it. First restriction enzyme is what? HIND2. Another thing in this particular chapter I just want to say is source of restriction enzyme. Source of restriction enzyme. What is the source of restriction enzyme? So answer of this particular question is which one? Bacteria. We are isolating these restriction enzyme from which one? Bacteria. Next potential question is why bacteria produce RE. I am just talking about the hits area. Produce restriction enzyme. Answer this question. For which purpose? Answer this question. Defense. With the help of restriction enzyme, bacteria perform defense against bacteriophage virus. Bacteriophage virus. Is it clear? Now if we just move ahead, how they perform the defense against bacteriophage virus, I am just going to tell about this paragraph in a brief from where question is going to ask. Suppose that this is a E. coli bacteria. Is it? It is a E. coli bacteria. And here it is a virus. It is a bacteriophage. This is a bacteriophage. This is the genetic material of the bacteriophage. And this is the genome of the bacteria. Right? Now this virus, what this virus basically do, this virus incorporate their genetic material inside the bacteria. Suppose that it is a viral genetic material. Is it clear? So they incorporate their genetic material in the virus. Then what happen? If these bacteria not going to manage it, what happen? These virus population going to increase inside the bacteria, and bacteria get infected by virus. So what bacteria is doing? These bacteria releasing two type of enzyme. Two type of enzyme, or two different enzyme, you can say. Two different enzyme released by bacteria. Here I am somehow slow because from here I just want to explain something. So two different enzyme produced by this bacteria to protect itself from the infection of virus. So one enzyme perform methylation of bacterial DNA. Methylation of bacterial DNA. First enzyme perform methylation of the bacterial DNA. Getting the idea or not? They are adding methyl group on the bacterial DNA. Methyl group, simply you keep in mind. Why they perform methylation of the bacterial DNA? To protect its own DNA from RE, restriction endonuclease. And what done by the second enzyme? Second enzyme cut viral DNA into pieces. So how they protected themselves? They are releasing an enzyme, restriction endonuclease enzyme. These restriction endonuclease, what doing? They are chopping the viral DNA, and from this enzyme, this methyl group protecting the bacterial DNA. So with one side they are saving their own DNA, another side they are cutting the viral DNA. So for this purpose, bacteria release restriction endonuclease enzyme. Got the idea? So first enzyme, what the name? One student wrote also, this is methylase. And second enzyme we are saying is which one? RE, restriction endonuclease. You got the idea? It is restriction endonuclease. Clear? So I hope this paragraph is done. Over. Now we are just going to talk something more about the restriction enzyme. Restriction endonuclease. So these restriction endonuclease cut the DNA at a specific site. And if you see the recent trend of the exam, they are asking about these specific site. These specific site are palindromic. These specific site are what? Basically palindromic. What is this palindromic? Same in forward and backward direction. So we don't have more time for background and introduction. I am writing a palindromic sequence. GAA TTC GAA TTC. Got the idea? Here it is the palindromic sequence. So here it is 5' to 3', 3' to 5'. Now this particular recognition site is not cut by any restriction enzyme. It cut by a specific restriction enzyme, and the name of that restriction enzyme is EcoR1. EcoR1. No all enzyme can cut this sequence. So what happened? A particular restriction enzyme first inspect the recognition site, and if they are finding it is a correct recognition site, then these restriction enzyme act. So EcoR1 is going to inspecting this, and they are finding this is the enzyme for what I made. So what they are doing, they are cutting the DNA just away from the center. Here it is the center. So they are not cutting in the center, they are cutting away from the center. So So when it is cutting away from the center, but between same nitrogenous bit, here cutting between G and A, here also cutting between G and A. So after cutting, what happened? You will find they are going to form a such kind of a structure. Space is not downside, so I am writing. It is in word format open, so we can't scroll even. So have a look here. After cutting, it look like G, C, T, T, A. Another one look like, which one? A, T, T, C, G. Got the idea? So you just look, after cutting it looking like this. So what you are finding, what are the advantage of cutting away from the center? The advantage here is, after cutting, a single stranded overhang structure is there. This is single stranded overhang. So these single stranded overhang region facilitate the joining of two different DNA together. Single strand, suppose that vector DNA will come. So they go for a complementary pairing and they join by the hydrogen bond. And finally, they are going to join with the help of which one? DNA ligase. So I am writing here two terminology. It is popular one I'm going to ask in exam. One is molecular seizure, examiner will ask. Molecular seizure. I hope everybody know the name. Oh. Molecular seizure. Who is molecular seizure? Restriction endonuclease enzyme. Restriction endonuclease enzyme act as a molecular seizure. It cut the fragment of the DNA. So, got the idea? This is a molecular seizure. Example of molecular seizure. This is restriction endonuclease enzyme act as molecular seizure. It is cutting the DNA like this. Got the idea? But not anywhere in a specific region, that is called recognition site and palindrome. And what is molecular glue? Molecular glue. Who act as glue? Who act as joining enzyme? So it is ligase. So ligase act as a glue and restriction endonuclease act as a seizure. Keep in the mind. Now we are moving ahead. Now sometime examiner asks some question from nomenclature of the restriction endonuclease enzyme. So I am writing about the nomenclature with the help of example. EcoR1 is one of the enzyme, because more than 900 enzymes are there. Restriction enzyme is how many? More than 900 are there. So we have to make a frame a rule to giving the name. So first letter of an enzyme, E here. So E coming from, it is representing the genus of the bacteria. So Escherichia. Two letter in a small letter, CO, coli. So genus name in capital letter. Two letter for which one? Species name. It is in a small letter. So Escherichia coli, R stand for strain of the bacteria, and one is what basically? Order of enzyme isolated from a specific strain. Isolated from a specific strain of bacteria. So one here means it is the first enzyme isolated from Escherichia coli. So these are the method of giving the name of the nomenclature of the restriction enzyme. Got the idea? What I told about the nomenclature? First letter genus, second, third letter species in small letter, R is strain, and this is what basically order of enzyme isolated from a specific bacteria. Is it clear in the mind? Now I am moving ahead. In the cloning vector and screening part I am going to talk. As I told you, we are not going to say everything. So I am talking about a cloning vector and a screening part. So just give your attention. You will understand it. I will not going to put a lot of condition here. Simple basic things I am going to tell you. So you will understand that clear. Here it is tetracycline resistance gene. I am taking which one? PBR 322. 322 plasmid we are taking. So it is tetracycline resistance gene. It is a selectable marker. It is a selectable marker. From here question is going to ask, so be careful, listen it. And many of you stuck here also, I know it. It is ampicillin resistance gene. It is ampicillin resistance gene. So if you talk about the NCERT have shown 2 recognition site in tetracycline gene in between and 2 recognition site in between ampicillin resistance gene. Got the idea? Now just give your attention. Suppose that I am taking which enzyme? BAM-H1 enzyme. To cut the plasmid and vector DNA both. BAM-H1 to cut both vector DNA and desired gene. So when we cut the vector DNA with the BAM-H1, it will cut at which recognition site? BAM-H1 recognition site. So this is the recognition site for BAM-H1. Got the idea? This is the recognition site for BAM-H1. So when we are cutting with BAM-H1 restriction enzyme and joining the ligase, after that what we are adding? So we are adding BAM-H1, another thing we are adding which one? Desired gene. Another thing what we are adding? Ligase. How many possibility will arise? So 2 possibility will arise. Give your attention. Possibility number 1, when we adding which one? BAM-H1, desired gene and ligase enzyme. One chances is self-ligation occur. Means both sticky end of the vector DNA rejoin together. Meaning of what? Sticky end of vector DNA rejoin. So in this case, who will produce? Non-recombinant will produce. Means we cut it here by the RE but join with the ligase. So no changes occur in the tetracycline resistance. Second possibility is which one? Ligation of desired DNA occur with vector DNA. So here recombinant will produce, or I am writing. So I am just telling non-recombinant have resistivity for both ampicillin and tetracycline. Because we try to add a new gene, we not added. So no changes occur in the plasmid, original makeup of the plasmid. So non-recombinant have resistivity for ampicillin and tetracycline both. Now listen me carefully. If we are cutting the vector DNA and desired gene with BAM-H1, if new gene not added, non-recombinant have resistivity for both ampicillin and tetracycline. If new gene added, recombinant is forming. Recombinant have ampicillin resistivity, but no resistivity for tetracycline. Why tetracycline resistivity is lost in recombinant? So we add the new gene between the old gene. Here we added a new gene between the old gene. Old gene is tetracycline resistance gene. Got the idea? And so new gene when added between an old gene become inactive and it is called insertional inactivation. Are you getting the idea or not? So the student who are stucking more, for them I am writing some line. It will help you at least in the exam. If BAM-H1 enzyme used, right? In that case, ampicillin resistance gene help to differentiate between, simply keep this one in mind, transformant and non-transformant, T and NT, and tetracycline resistance gene help to differentiate between recombinant and non-recombinant. If you keep these things even in mind, in NEET, there is a chance you can handle the question. But condition is there. If we are using which enzyme to cut? BAM H1 to cut. NCERT use BAM H1 to cut. So many times what happened, paper setter even not give the name of enzyme. In that case, you consider it what? BAM H1, because NCERT wrote BAM H1. If enzyme name is not given. But most of the time they give which enzyme we are using. So if BAM H1 using, ampicillin resistance gene help to differentiate between transformant and non-transformant. So ampicillin resistance gene help to differentiate between transformant and non-transformant, acting as a selectable marker, and tetracycline resistance gene help to differentiate between recombinant and non-recombinant. Ampicillin between T and NT, transformant and non-transformant, tetracycline resistance gene between recombinant and non-recombinant. The student who are studying more for them, right? Now I am moving ahead. So now I am going to tell about blue-white screening. Blue-white screening. So what happened in blue-white screening? Here there is a plasmid, vector DNA. Try to understand. It will take 1 minute. We will fix it also. 1-2 minute maximum. So this is the plasmid. In this plasmid, here it is a selectable marker gene. Selectable marker gene. Example is LAC-Z gene. LAC-Z gene is a selectable marker. Selectable marker gene. Got the idea? Now, if we talk about here, in this LAC-Z gene, in between this LAC-Z gene, there is recognition site for a restriction enzyme. It is not mentioned in NCERT for which restriction enzyme. Now I am taking restriction enzyme because NCERT not mentioned, plus desired gene. It is seizure, it is another glue ligase. Then RE will cut here. If it will cut, there is a chances of desired gene may join and self-ligation occurs. So here also how many possibility exist? 2 possibility. One, who will produce? Non-recombinant. Another, who will produce? Recombinant. So in non-recombinant, for desired gene not adjoin. Desired gene not ligated, not join with vector DNA. And in case of recombinant, desired gene join. Desired gene join. Got the idea? Understand the idea? So if desired gene not join, no any new gene added here. So LAC-Z gene remain functional or not functional? So in case of non-recombinant, LAC-Z gene remain functional because no new gene added. In non-recombinant, it functional. Why it functional? Because we try to add a new gene, but it not added. Self-ligation occurs, self-joining occurs. And in recombinant, what happened? LAC-Z gene or gene for beta-galactoside, we also say, become inactive. Got the idea? Remain inactive. Why remain inactive? Due to insertional inactivation. Due to insertional inactivation. Did you got the idea? Due to insertional inactivation. What is insertional inactivation? When a new gene added between an old gene become inactive. So here we added a new gene here. So this old gene become inactive. Got the idea? So in case of recombinant, LAC-Z gene become inactive. So non-recombinant produce which enzyme? Beta-galactosidase. Because lag Z gene produce which enzyme? Beta galactosidase. You better know, na? Or it is gene for here lag Z gene when inactive, no beta galactosidase. No beta galactosidase. Did you got the idea? No beta galactosidase. So in non-recombinant, who is producing? Beta galactosidase is producing. In recombinant, beta galactosidase is not producing. No beta galactosidase is producing here. Here it is no beta galactoside. Here beta galactoside is producing. Now how to check beta galactoside is producing or not? So we are adding chromogenic substances plus chromogenic substances. We are adding a chromogenic substances. So when chromogenic substances added with beta galactosidase is term which color? Is term which color? Blue color. And here no beta galactosidase is there. So when we add the chromogenic substances, it not give blue color. So it is a colorless colony or white colony. Got the idea? So simply by identifying the color, if non-recombinant is there, beta galactosidase present, it react with chromogenic substances giving blue color. If case of recombinant, what happened? Lag Z gene not produce beta galactosidase due to insertional inactivation. When we add chromogenic substances, color will not change and it remain white color. You got the idea? So this is the blue-white screening from where questions is also going to ask. I hope you understand it. Can we move ahead? So simply for the last moment, non-recombinant blue, recombinant white in presence of chromogenic substances. Done? Now I am moving ahead. Now give your attention. I am just giving you a basic idea about from where question is going to ask PCR, polymerase chain reaction. So rather than to draw, take the help of textbook. Electrophoresis I will also say, don't worry. So we are going to talk about polymerase chain reaction. Okay, ji? So now give your attention about polymerase chain reaction. See, first PCR is a method of gene amplification in vitro. Got the idea? So what is this? It is a method of which one? Gene amplification. PCR is a method of which one? Just a minute, give me a moment. So polymerase chain reaction is a method of gene amplification. Method of gene amplification in vitro. It is slow. Method of gene amplification. So how many steps of PCR and what is the correct sequence of step? It is going to ask an exam. First step is denaturation. Denaturation, what happened in denaturation? At high temperature around 92 to 94 degrees Celsius, the DNA strand get broken. Just see hydrogen bond going to broken. So DNA get denatured. Breaking of H bond occur, that is called denaturation. That occur at high temperature. So this is the very first step during PCR is denaturation. Every year people ask this question. Denaturation occur at high temperature. Right, that is 92 to 94 degrees Celsius. Now, when both the strands get separated, we are performing annealing. Annealing means cooling. So cooling, annealing means cooling. So at which temperature? 56 degrees Celsius. And after cooling, what we are adding here? We are adding the primer. DNA primer we are using. Got the idea? DNA primer we are using. Getting the things or not? It is RNA primer or DNA primer. So keep in mind during PCR we are using DNA primers. How many primer we are using? 2 primer. And primer we always add in which direction? 5' to 3' direction. This event is called annealing. You got the idea? Now we are going to talk about the third step, that is extension. Extension we perform with the help of an enzyme Taq polymerase. It is a type of DNA polymerase isolated from Thermus aquaticus bacteria. That bacteria sustain at high temperature. Now you are saying ki why we are taking this especially Taq polymerase, sir, normal Taq polymerase, why I am not taking? So because we are doing denaturation at very high temperature, and we are adding everything in a machine, na? Getting the things or not? So we are adding everything in a thermocycler machine, right? Nucleotide, primer, DNA, enzyme, and that machine is doing. So these denaturation occur at this high temperature, and at that high temperature if we add which one? A polymerase who don't have stability at the high temperature, they will denature. So that is the reason we are taking it from which one? Thermus aquaticus. Cutting the things or not? Right? So this is Taq polymerase. They are performing polymerization or extension. So from one DNA, two DNA is formed here. And from one gene, two genes are forming. If we are going to repeat for 30 cycles, it is going to produce approximately how many million copy? 1 billion copy going to produce. So what are the correct sequence of a step? Denaturation, annealing, extension. And in such a way we can amplify a number of a particular gene into billions of copy. So these are the polymerase chain reaction. In zoology side, probably your sir told, it have used in diagnosis of the diseases as well. Got the idea? Now we are moving ahead. So now I am just going to say if 1-2 line about downstreaming. Sometimes student ask about it. What come under downstreaming process? In downstreaming process, what happen? Everything start from isolation of product to before marketing called downstreaming process. Suppose that in bacteria you produce recombinant insulin. Insulin now in the bacteria. So insulin forming as which one? A polypeptide chain and B polypeptide chain. So you are removing A polypeptide from one bacteria, isolating it, B polypeptide from another bacteria. So isolation comes under the first event. Got the idea? Understand? Isolation of A polypeptide and B polypeptide. This is the first event of the downstreaming process. Example I am telling you. Then after isolation you are purifying it because we are isolating the protein polypeptide, some an impurity, another polypeptide also came. So you are going for purification of it. Purest form you will take. So purification, isolation, after that purification. Now you are adding A polypeptide with B polypeptide by disulfide bond. Then it become a which one? Insulin. So that changes you did. So you made that insulin now. Now what happened? You are sending this insulin for trials and testing. So trial and testing done. Government agency giving you the license, right? After licensing, what you are doing? You are producing the product in the large scale in a bioreactor, and then you are adding a preservative and deciding the expiry date. So every event from isolation up to here, it is called which one? Downstreaming. Marketing not a component of downstream processing. Got the idea? So these are the key area from this particular chapter. Now we are going to talk about the next chapter. Give your attention. Anything you want to from this particular chapter, you just put it. Anything more you want from here? Is it done? Now I am moving ahead on the next chapter of the biotechnology. Why I started from biotechnology? The reason is from this chapter, a lot of questions is going to ask in the NEET. And especially you be careful about the PCR. So now I am coming on the second chapter of the biotech. Biotechnology and application. Here I will just going to talk about the two area. Hanji. So in biotechnology and its application, we are just talking about which one? BT. Just listen first. Two area we will talk, RNAi, BT and full form of GEAC. So one area BT. BT stand for Bacillus thuringiensis. It is a soil bacteria. Got the idea? Bacillus thuringiensis is a soil bacteria. And these Bacillus thuringiensis have a gene that is called CRY gene. CRY gene. These CRY gene in bacteria, Bacillus thuringiensis, produce a protein called CRY protein. CRY protein. Or full name of this protein is crystalloid protein. What is the full name? Crystalloid protein or crystalloid protein. Or crystalloid protein. Got the idea? Now, this crystalloid protein not produced by bacteria every time, produced in certain stage of life. Produced in certain stage of life. Is it clear? Produced in certain stage of life. Okay. Now what happened? These crystalloid protein present as protoxin in bacteria. Present as protoxin. Protoxin is inactive or insoluble. Insoluble and inactive. So when this protoxin producing in bacteria, it remain inactive. Got the idea? It remain inactive. And when it remain inactive, it not harming the bacteria. Now what happened? Now this protoxin in midgut of insect. In midgut of insect. In midgut of insect, what this protoxin is doing? They converted into toxin. Become toxin. Why it become P toxin? At alkaline pH. At alkaline pH it become toxin or it becomes soluble. And when it becomes soluble and it becomes toxin, it bind with epithelial of midgut. It bind with epithelium and create pore. In food pipe, pore created. Got the idea? Through that pore who enter? Water enter, it gets swell and die. So insect get die. So these are the basic working of these particular crystalloid protein. Now it is how it is helping us in genetic engineering. Let me tell you how it is helping us in genetic engineering. Getting the idea or not? Right. So just a moment. How it is helping us. Now scientists did what? Suppose that we want to make BT cotton. BT cotton is a which type of plant? It is a genetically modified organism, or we also call it transgenic plant. Transgenic plant. Got the idea? So these BT cotton, normal cotton have what problem? Cotton get attacked by, infected by which one? Bollworm. Got the idea? So when bollworm infecting, bollworm is an insect. Got the idea? It is not a worm, it is an insect. And these bollworm reduce the yield of cotton. It is insect pest. Got the idea? So it reduces the yield. This is the problem of the cotton plant. Now what we did in the BT cotton, we have taken cry-gene from the Bacillus thuringiensis of BT, added with a vector DNA. Suppose that this is a vector DNA and is it TI plasmid. Assume TI plasmid. It is oocyte. Now what we did, we cut the vector DNA, and after cutting the vector DNA, we attach the desired gene and making a rDNA, recombinant DNA or chimeric DNA. Recombinant DNA is also called chimeric DNA. Now I have taken a cotton plant cell and introduced this one into the cotton plant cell. Now through tissue culture, we made a plant cell, entire plant. Not a single plant, we make billion of plant by micropropagation. Getting the things or not? Many plants are there. Now it is a GMO, genetically modified which one? Cotton. Genetically modified cotton. Now larva of bacillus, larva of which one? Bollworm is coming. Caterpillar or larva of bollworm is coming and feeding in the plant part. So plant cell will enter into their food pipe, and in their food pipe along with the plant cell, in every cell who is there? Cry protein is there, crystalloid protein going to release here in the gizzard when cell will break, then crystalloid protein enter in the midgut. So in midgut at alkaline pH they are binding and creating a hole. Through that hole water is entering, swelling occur, lysis occur and death occur. Got the idea? So in such a way we just utilize entire of this particular mechanism. Understand the idea? Is it clear? Is it all right? So how we are making BT cotton in such a method way, we are making BT brinjal, BT rice, BT soya bean. Got the idea? So many different variety of the Bacillus thuringiensis. Now we are just so many genetically modified organism by using BT gene. Is it clear? Now we are moving ahead. Two gene you don't forget in mind, Cry1AC and Cry2AB. These two genes provide resistivity against which one? Cotton. Directly this line can come in exam. Cotton bollworm. Don't forget the name of this gene. Cry2AB, Cry1AC. And Cry1AB gene provide resistivity against corn borer. Now I am asking a question. I am asking a small question. Is it possible for cry gene to kill all insect? Yes or no? No. Now I am asking another question. Which order insect killed by this crystalloid protein? Write the name of order with example. Write the name of order. So how many order given in NCERT? Lepidoptera, Coleoptera, Diptera. Got the idea? So they are insect group specific. Coleoptera, Lepidoptera, Dipteran insect. These three category are there. You don't forget about these insect category. Got the idea? So this one is over. Now we are moving ahead. These things you please don't forget. What I am underlining, Okay. Bacillus thuringiensis produce protein that kills certain insects such as lepidopterans, that is tobacco birdworm, armyworm, coleopterans, that is beetles, and dipteran, flies and mosquito. If I ask bollworm belongs to, you will also write here bollworm. Bollworm also belongs to which one? Lepidopterans. Tobacco birdworm, armyworm, you also write in this list as what? Bollworm. So these are killed by coleopteran, beetles and dipterans, flies and mosquito. This is a potential line to ask a match of the following type of question. Got the idea? So don't forget this. It is over now. Now we are moving ahead. Give your attention. Now we are moving toward RNAi. Just give your attention. RNAi. I am just telling about RNAi in a board directly. No need to RNAi. I am telling 5-6 line and 99% chances are there from RNAi question is going to ask from this line only. Got the idea? Right? So in RNAi, I stand for interference. Interference. So in RNAi, I is what basically? Interference. Question I am asking, what is RNAi? 5-6 question I am telling. What is RNAi? Answer of this question. It is a method of cellular defense. Cellular defense in eukaryote, not in prokaryote, by post-transcriptional gene silencing. By post-transcriptional gene silencing. Because it is doing the gene silencing at post-transcriptional level. Post-transcriptional gene silencing not occur in prokaryote, it occur only in eukaryote. So what is RNAi? It is a method of cellular defense in eukaryote. How? By post-transcriptional gene silencing. Got the idea? Second, nematode infect. Answer. Plants, animals, fungus, fungi and human all get infected by nematode. Got the idea? Now I am going to talk about MI, Meiosis incognitia. It is a nematode. Meiosis incognitia is a nematode. These nematode infect which one? Infect root of tobacco plant. It infect root of tobacco plant and reduce the yield. Reduce the yield. Now what we did with the help of RNAi, so with the help of RNAi, techniques, got the idea? We developed a transgenic tobacco variety. Transgenic tobacco variety. What the idea? Earlier, who is killing whom? Earlier, nematode was, Meiosis was killing the, which one? Tobacco plant. Now, transgenic tobacco plant is just reverse. Now, if Melidogyne incognitia came and fed on root of the tobacco plant, who will die? Nematode will die. Melidogyne will die. Earlier plant was dying, now nematode will die. How? So another question. During RNAi, what is the source of nematode specific gene? This is a very important question. Nematode specific gene. So first is two source NCERT wrote. One source is RNA virus, which nematode specific gene or complementary RNA. Got the idea? RNA virus which replicate via RNA intermediate. First source. And second source is transposon. This is a mobile genetic element. These two used as a source during RNAi. NCERT wrote like this. Got the idea? Vector used during RNAi. So agrobacteria. Agrobacterium used as a vector during RNAi. Did you got the idea? Is it clear? Now NCERT wrote कि we introduce, I think page is smaller here. We introduce, I will explain it, don't worry. Nematode a specific gene into host cell in such a way it produce double-stranded RNA. Got the idea? It produced the double-stranded RNA. Now try to understand the picture. I am giving you the details in 1 minute. Suppose that this is nematode specific gene. But we want to introduce this gene in host cell in such a way it produces double-stranded mRNA. So for that purpose, we are attaching both side promoter. So both side RNA polymerase will bind. Now I am taking a TI plasmid from agrobacterium and I am adding this nematode specific gene with this. So it become like this. It become like this. Just give your attention. It is recombinant DNA. Now I am taking a cell of tobacco plant. I am incorporating it here. And from here now, this is a nematode specific gene enter now into which one? Tobacco plant cell. Now with the tissue culture, we are making a entire tobacco plant. Tissue culture, we are making a tobacco plant. It is tobacco plant. But what are unique in this tobacco plant? Every plant cell have which gene? Nematode specific gene. Every tobacco plant cell have nematode specific gene. Now what happened? In every tobacco plant cell, what happened? In every tobacco plant cell, I am just dealing according to NCERT. Otherwise, detailing is somehow different. In every tobacco plant cell, these nematode specific gene, nematode a specific gene going for transcription and making dsRNA. It is a dsRNA. So it is called sense strand 5' to 3' and 3' to 3'. Getting the things or not? It is anti-sense strand. Now what happened? Now Meiosis eating the tobacco cell. So this is the food pipe of Meiosis. And this is the cell of the Meiosis. And this is a vital gene of Meiosis. If this gene will not express, this cell will die. Got the idea? This is a vital gene of Meiosis. Got the idea? Now this Meiosis producing which one? mRNA. And these mRNA producing which one? Protein. These protein acting as a catalyst, converting a substrate into product, reaction going on, and this product is essential for survival. Essential for survival of Meiosis. Survival of Meiosis cell. Now what happened? When this cell enter into the food pipe, cell get rupture. And according to NCERT, I am telling every time, according to NCERT, this is double stranded mRNA. dsRNA. It is, got the idea, sense or it is anti-sense strand. So in actual what happens, sense strand digested. And these anti-sense strand bind with which one? mRNA. They enter from the alimentary canal, food pipe to cell, and they bind with the mRNA, make a double stranded mRNA here. Protein synthesis stop. If protein synthesis stops, substrate not convert into product. These essential product will not form who require for survival of the cell. Cell will die. If many such cell will die, nematode will die. Got the idea? So what key question going to ask from here, I am just telling you. First, what is RNAi? Method of cellular defense in eukaryote. Second, am I infect? Root of tobacco plant. Third question, source of nematode specific gene during RNAi, RNA virus or transposome. Next question, vector used during RNAi, agrobacterium. Next, how we introduce nematode specific gene into the host cell in such a way it produce double stranded RNA. Out of double stranded RNA, one is sense strand, another is anti-sense strand. Got the idea? Now these double stranded RNA, according to NCERT, bind with the mRNA of nematode and stop their expression. If mRNA of nematode will not express, that cell will die. If many such cell will die, nematode will die. So this is about the RNAi. Got the idea? At least these information you keep in mind. It will done. Okay, Ji, over. Can we move ahead? So another thing, GEAC. Tell me the full form of it. Every alternate year this line going to ask an exam. GEAC. What is GEAC? Tell me. Genetic Engineering Approval Committee. This committee in India look after safety related to GM research and GM product. Got the idea? This look after Genetic Engineering Approval Committee look after the safety related to GM product and GM research. Now something you must keep in mind, I think everybody know, like golden rice. It is a genetically modified organism and it is rich in vitamin A. We are adding two different genes in the, which one? Golden rice, one from daffodil, another gene we are adding from a soil bacterium. To produce which one? Beta carotene. Is it clear in the mind? Right, very good. Now we are going to move ahead. Biotech is over. Now I am going to tell about the microbe 3-4 line. Then we will come on molecular basis of inheritance, getting the things or not. So give your attention. Have a look. So I am saying microbes first because reading NCERT is somehow painstaking job. It is going to slow our speed. Okay, so let's tell me. So listen it carefully. It will help you. Curd is produced by which bacteria? Lactobacillus, lactic acid bacteria. Curd is superior food than milk because in curd who is there? Vitamin B12 is there. Vitamin B12 is there and another organic acid are also there. Got the idea? Butyric acid. Which bacteria? Pin your answer. Butyric acid. Which microbes are producing butyric acid? Fast. Butyric acid. Clostridium butyricum. Very good. Citric acid. Citric acid. Citric acid produced by which bacteria? Fast. Aspergillus niger. Very good. Baker and brewerist. Saccharomyces cerevisiae. Baker and brewerist is what? Saccharomyces cerevisiae. Is it? Done? Right. Now we are going to talk about Swiss cheese produced by which one? Swiss cheese. Swiss cheese. Propionibacterium sermani. Roquefort cheese. Penicillium roqueforti. Roquefort cheese by which one? Penicillium roqueforti. So these are the some of the bioproduct, useful product, and these are the name of microbes. But the most important one is, you please see here. From here, there is a possibility of asking a line, a question. That's the reason I am just telling you. Chemical enzyme and this paragraph, it is important. So you please don't forget about cyclosporine A. Don't forget about cyclosporine A. It is a bioactive molecule used as immunosuppressive agent in organ transplantation in patient. Got the idea? So produced by which one? Trichoderma polyesporum. Don't forget these things. Three things going to match up the following is a potential question from here. Cyclosporine A is act as immunosuppressive agent that required during transplantation of organ. Sources of this particular cyclosporine A is Trichoderma polyesporum. Got the idea? Another, a streptokinase. Got the idea? It is produced by a streptococcus bacteria and this is used as clot buster. Don't forget this line. Don't forget this line. Got the idea? Another one is a statin. It is produced by yeast. What is the name of yeast? Monoscus purpureus. And this used as blood cholesterol lowering agent. Blood cholesterol lowering agent. Who is the statin? So you have a high level of cholesterol, you use which one? A statin. Got the idea? I just telling it for what, which purpose? Mugging up purpose. So don't use it without recommendation of doctor. So statin is doing which particular job? Reducing the cholesterol level. It is isolated from which one? Yeast Monoscus purpureus. Another thing is what basically in laundry industry, we are using which enzyme? Lipase enzyme. It is removing the oil stain from your cloth. So enzyme lipase are used in the laundry industry and juice making industry. Which enzyme is going to use? Do you know? Pectinase is going to use. Pectinase is going to remove the fiber from your juice and juice become transparent, as well as yield of the juice is going to also increase. So these are a very, very, very important paragraph for asking the question from the microbes. Now I am just telling you about the sewage treatment in 1 to 3 minutes. Got the idea? So have a look about the sewage treatment. So according to NCERT, we are going for 2-stage treatment of the sewage. How many stage treatment? Okay, let me write, otherwise satisfaction not came. Haji. So in sewage treatment, sewage is what? Basically domestic waste. Let me write the key point from here. It is going to ask sometime. So 2 stages of treatment are there. I'm not going to tell you the entire process. I'm going to finish in 2 minutes, 3 minutes, whatever the time it requires. One is primary treatment. Write it short. Another is secondary treatment. So in primary treatment, 2 steps are there. Filtration. One step is filtration, another step is sedimentation. And sedimentation. Primary treatment is a physical process. So, what are the solid waste? Solid, large, non-dissolved waste. We are removing by filtration. Bottle, tin, polythene, plastic, shoes, big stone, they are going to remove by filtration process. Now sedimentation. In sedimentation, what happened? We are transferring this particular sewage in a sedimentation tank. So in tank, what happened? Some suspended waste are there who are going to settle down. Suspended waste are going to settle down. It is called primary sludge. Primary sludge. Who are settled down in this one? These primary sludge consist of clay, pebbles, sand. Got the idea? Small stone, very small stone. And what is floating here? Not settled. In sedimentation tank, who is floating is called effluent. These effluent consist of 2 components. Water, 3 components you can say. Bacteria plus dissolved organic waste. Dissolved organic waste. So we remove this big solid like bottle, tin, cloth, plastic through filtration. We filtered clay, pebbles, sand, small stone through sedimentation. Now we are transferring this effluent into for which one? Secondary treatment. So secondary treatment I am just writing. In secondary treatment, what we are doing? First we are transferring this into which one? Aeration tank. Aeration tank. So this is aeration tank. This is aeration tank. In aeration tank, what we are doing? We are transferring the effluent. So in effluent, who is very high? BOD is high. Why BOD is high in effluent? Because of dissolved organic matter. Organic waste. This is the major challenge for us. Dissolved organic waste is there. Due to that reason, BOD is high. BOD is indicator of pollution. More BOD, more water pollution. More water pollution. So now what happened? In aeration tank we supply air. There is a agitator, mixer who mix everything. So when we supply air, the population of aerobic bacteria increase dramatically. Aerobic bacteria plus fungi, their population increase, and they aerobic bacteria and fungi start consuming organic waste. They are heterotrophs. They are getting the food or energy from which one? These dissolved organic waste. Slowly, after a time, who decrease? BOD level decrease. As BOD level decrease, what happened? We are shifting this entire things in another chamber, another settling tank. So this is another settling tank. We are discharging the low BOD waste. So who settled in the bottle is called activated sludge. What is the composition of these activated sludge? Flocks. Because these aerobic bacteria plus fungi is also called flocks. And who are effluent is what? Water with low BOD. Low BOD. Be discharged into the river. And what we do with this particular activated sludge? So some of the activated sludge we are taking as a inoculum and transferring into aeration chamber, and most of them we are transferring into an anaerobic sludge digester. In these anaerobic sludge digester, who is there? Methanogen. And these methanogen produce which one? Biogas. And what are the composition of this biogas? CH4, CO2, H2S. Got the idea? So this is about the sewage treatment in a brief. You got the idea? I hope you understand. Now from this chapter, another thing is important is biocontrol agent example. Just a minute. Biocontrol agent example. These examples are important. These are going to ask question from here. Got the idea? This paragraph is highly important. I am underlining it also. So you please don't forget about the lady word and dragonfly is a biocontrol agent against aphids and mosquito respectively. Means lady word is against which one? Lady word is against aphids. Got the idea? And dragonfly is against which one? Mosquito. So if population of dragonfly is high, whose population will under control? Mosquito population remain under control. If the population of ladybird is high, dragonfly, which one? Aphid population remain under control. So ladybird against aphids and dragonfly against mosquito. Got the idea? Don't forget this. Another biocontrol agent is Bacillus thuringiensis. In market, it came in a pouch, sachet. Farmer purchase it, mix the Bacillus thuringiensis with the water and spray on the plant. These bacillus present on the leaf surface. When any insect of a specific group came and feed them, same problem occur, create pore in the midgut and they kill the insect. Got the idea? So in that way, we are able to manage the larva of which one? insect like butterfly, getting the things or not? Now we are going to talk about which one? Trichoderma, third biocontrol agent. 2 we already discussed, 3 we already discussed, ladybird, dragonfly and BT. Now fourth we are going to talk about trichoderma. It is a biocontrol agent against which one? A biocontrol agent being developed for use in treatment of plant disease is the fungus trichoderma. Fungus trichoderma used as a biocontrol agent to protect from various plant diseases. Trichoderma species are free-living fungus that are very common in the root ecosystem. They are effective biocontrol agent for several plant pathogens. It protects the plant from the pathogen of the plant if this trichoderma fungi is present in the root. Another one is which one? Baculovirus. Baculovirus is a biocontrol agent against insect and other arthropod. What the idea? It is a kind of nucleopolyhedrovirus, and these are a species specific and narrow spectrum insecticidal. Broad spectrum means who kill variety of insect. Narrow spectrum means who kill a specific insect. Got the idea? So 5 example of biocontrol agent is there in the NCERT. First, ladybird against aphid. Second, dragonfly against mosquito. Third, BT against insect caterpillars, got the idea, and larva. Other one is which one? Trichoderma against root-borne pathogen. Another example is which one? Baculovirus, that is a nucleopolyhedrovirus, and it is a narrow spectrum, and it used as a biocontrol agent against arthropods and insect. Please don't forget these 5 examples. There is a potential to ask question from this particular paragraph. Got the idea? Is it clear? Done? Can we move ahead? Haji. So now we are just going to say something about biofertilizers. Got the idea? From here you can also expect questions. So biofertilizer usually used by organic farmers. Used by which one? Organic farmers. Got the idea? They are not using chemical fertilizers. So okay, now we are just going to talk. All of you are familiar about rhizobium. They perform symbiotic nitrogen fixation in leguminous plant. Got the idea? Is it clear? Right? So other examples of biofertilizer given is other bacteria can fix atmospheric nitrogen while free living in the soil is azospirillum and azotobacter. Don't forget this. Right? What they are doing, they are enriching the nitrogen content of the soil. Azospirillum and azobacter. Rhizobium is a symbiotic. Got the idea? Now you don't forget, is the most important from here. So fungi also known to form symbiotic association with plant. You know about mycorrhizal association. Got the idea? So many member of genus Glomus. Genus Glomus is a genus of fungi, form mycorrhiza. The fungal symbiont in this association, what work done by the fungal partner? Examiner asks. They absorb phosphorus from the soil and pass this phosphorus to plant. Plant having such association, so other benefit also. What are the other benefit? So such as root resistance to root-borne pathogen. These mycorrhizal association protecting the plant from root-borne pathogen. They provide tolerance to salinity and drought. Got the idea? And overall increase in the plant growth and development they are playing. Got the idea? So these are the advantage of fungal partner. Fungal partner absorbing the what? Phosphorus. They are protecting from which one? root-borne pathogen. They are responsible for increasing the tolerance of plant to salinity and drought, and it is involving in which one? Overall increase of the growth of the plant and development of the plant. Got the idea? Some of the cyanobacteria, it is written cyanobacteria are autotroph microbe widely distributed in aquatic and terrestrial environment, which may fix atmospheric nitrogen. Some of the example is what? Anavina, nostoc, oscillatoria, etc. In paddy fields, cyanobacteria serve an important biofertilizer. This exam line is important for exam. So in paddy field, who act as an important biofertilizer? Cyanobacteria, blue-green algae use as an important biofertilizer. Got the idea? Is it clear in the mind? So this is about the biofertilizer and this is about the chapter. Got the idea? Now two more line is there. Tell me the alcoholic drink produced without distillation. Alcoholic drink produced without distillation. Wine and beer. Remaining other alcoholic drink have what? Produced after distillation process. So who produce without distillation have alcohol content less than or equal to 13%. Chemistry book wrote how much? 14%. So deal according to the subject. So this chapter is also over. So 3 chapter done. Now we are moving toward the 4th chapter. Right. 3 chapter and 1.5 hour done. I think it is 1.5 hour, 3 chapter. Now we are coming on the other chapter, and that is, from where should I take first or second? Molecular biology, molecular basis of inheritance. Give your attention. Last year, 7 questions came from this chapter, molecular basis. That's the reason I am taking first that chapter from where we are usually finding more number of questions going to ask. Okay, ji. So come on the molecular basis of inheritance. I am just telling about the key area. My purpose is not to teach you the concept. Many times as a teacher we also become emotional, start saying something. Like in sewage treatment, I don't want to say that much, but I told you. Okay, right. Just give me a moment. Haji. So, in molecular basis of inheritance, what you please keep in your mind is, these are the data. See, for NCERT wrote DNA is a long polymer of deoxyribonucleotides. So number of nucleotide and base pair is variable from a species to a species. So it is also showing the unique property of a living organism. In every species, there are not a same number of base pair are there. Example is given. So what NCERT wrote here, there is a bacteriophage 5x174. It is single stranded. Got the idea? DNA. And how much nucleotide is there? 5386 nucleotide is there. Mind it, it is not base pair. It is single stranded. So how many nucleotide is there? 5386. This is the question going to ask in data. Match of the following. So 5x174, 5386. Lambda phage have, it is double stranded. How many base pair? 48502 base pair. Escherichia coli have 4.6 into 10 to power 6 base pair. Sometime examiner simply write 4 into 10 to power 6 base pair. It is also correct. Correct is what? 4.6, but sometime they wrote 4 into 10 to power 6. And human haploid content 3.3 into 10 to power 9. And if human diploid they ask, so diploid become which one? 6.6 into 10 to power 9 in diploid. In haploid it is 3.3. So these are the data, please you don't forget. Got the idea? Now I am just going to say about what type of bonds are present in the DNA. Just let me tell you. Just let me tell you about that. It will take 2 minutes. I am just removing it. These NCRT act as a speed breaker. Give your attention. Hanji. So I'm just giving a basic diagram and I'm going to tell you the type of bond present in the DNA. See, this is a double-stranded helically coiled DNA. I am just telling you about this. Just double-stranded helically coiled DNA given by Watson and Crick. It is a right-handed helix, right-handed DNA. It is B form of the DNA. B form of DNA. Got the idea? Right-handed helix, and it is one turn. It is one turn of the DNA. One turn. If we talk about the first type of bonding, so if you see, DNA is made up of strands and steps. It is 5 prime, it is 3 prime, it is 3 prime, it is 5 prime. So both the strands of the DNA are anti-parallel. Now to whom we are saying 3 prime, so when sugar have free OH at third position, it is called third prime. And when sugar have free phosphate attached at the fifth positions of the sugar, it is called 5 prime. And easy to understand, where phosphate is free is 5 prime, and where sugar is the last and sugar have OH is 3 prime. So 5 prime to 3 prime, 3 prime to 5 prime. So these are the both strand of the DNA. So strand of the DNA is made up of alternate arrangement of phosphate, sugar, phosphate, sugar, sugar, phosphate, sugar, phosphate. Now I am telling you about the bond. You just please keep in mind. So between sugar and nitrogenous base, N-glycosidic bond is there. N-glycosidic bond is there between sugar and phosphate. Between nitrogenous base, H bond is there. H bond is there. Got the idea? H bond is there between the nitrogenous base. Between adenine and thymine, double hydrogen bond. Between cytosine and guanine, triple hydrogen bond is there. Got the idea? Between sugar and phosphate, ester bond is there. Here, sugar and phosphate, ester bond is there. So one phosphate attached with 2 sugar, and this bond altogether 2 ester bond is called what? Phosphodiester bond. Phosphodiester bond. You don't forget 2-3 things. One thing, 2 nucleotide of same strand joined together by 3 prime to 5 prime, don't forget it, phosphodiester bond. 3 prime to 5 prime, phosphodiester bond. Another thing you don't forget is what? 2 nucleotide opposite strand. Joined together by hydrogen bond. joined together by hydrogen bond. You got the idea? So here you just try to understand with the example also. Look here, this is one nucleotide, this is another nucleotide, belongs to two different strands. In between who is there? Hydrogen bond is there. And two nucleotide, this is one nucleotide, this is another nucleotide. Which bond is there? Two ester bond is there. Hence we wrote it is what? Phosphodiester bond. You got the idea? Understand? So in DNA, how many type of bond is there? Three type of bond is there. One is glycosidic bond, another is hydrogen bond, another is ester bond. These three type of bonds are present in a DNA. Got the idea? Now if we talk about pitch of the DNA, pitch. Pitch or turn we are saying. One turn equal to 3.4 nanometer. In angstrom you can write or 34 angstrom. Got the idea? Number of base pair in one turn is equal to 10 base pair. Got the idea? So angle between two base pair is 34 divided by total angle. One turn equal to how much degree? One turn equal to 360 degree angle. See 360. From here to here 180 degree. From here to here another 180 degree. So one pitch or turn equal to 360 degree. Axial rise, angle between two base equal to 360 divided by 10 equal to 36 degree. And distance between two step here 3.4 angstrom or 0.34 nanometer. Diameter of the DNA 20 angstrom. Got the idea? Or in nanometer you can say 2 nanometer. You got the idea? These are the some basic about DNA. Now I am going to talk about Chargaff rule, from where question is going to ask. Chargaff rule. What is the Chargaff rule? So Chargaff told in a double stranded DNA, it applies only for a double stranded DNA. In a double stranded DNA, adenine is equal to thymine and cytosine is equal to guanine. It is not hydrogen bond, it is equal. What the idea? So in a double stranded DNA, adenine and thymine are equal and cytosine and guanine is equal. And these Chargaff rule also advocate the which one? Double stranded nature or complementary base pairing of which one? DNA. Now I am telling about, so we can say about, you better know na, in a adenine is a which one? Purine. Thymine is a pyrimidine. Cytosine is a pyrimidine. Guanine is a purine. Got the idea? So we are finding in a double stranded DNA who is equal? Purine and pyrimidine is equal. So we can say in Chargaff rule, in a double stranded DNA, purine is equal to pyrimidine. Purine is equal to pyrimidine. Purine is what? A plus G. Pyrimidine is what? C plus T. So in a double stranded DNA, what happened? A plus G upon C plus T is equal to 1. If this is equal to equal, so we can write like this or not. So this is the Chargaff rule. This is the Chargaff rule. Got the idea? Now, what happened? In a DNA, adenine plus thymine upon cytosine plus guanine is constant for a species and variable from a species to a species. It is not Chargaff rule. This is Chargaff rule. Many times students confuse. This is Chargaff rule. Adenine is equal to thymine and cytosine is equal to guanine. What B interpreted? B are interpreted purine is equal to pyrimidine. A plus G upon C plus T equal to 1. We are going for another interpretation. A plus T upon C plus G is a constant for a species and variable from species to species. How question is going to ask from the Chargaff rule? Examiner will ask a question. If I am asking only one question because we don't have much time, so I will say if a DNA, leave a if word, a DNA consists of two kilobase pair. 2 kilobase pair. If adenine is 15%, then find out the answer of following question. I am dealing this question because from here there is a possibility of ask the question. Got the idea? Question number 1. Percentage of cytosine. Second question is number of nucleotide. Third question, number of glycosidic bond. Fourth question, number of nucleosome. And fifth question, number of hydrogen bond. So I am just answering these all questions. Got the idea? One by one. So it will help you to handle the question based on this. Now I am telling DNA consists of 2 kilobase pair. 2 kilobase pair equal to 2 into 1000. Means 2000 base pair is there. 2000 base pair is there. Adenine is 15%. So if A equal to 15%, you better know A is equal to T. So T is also 15%. So adenine plus thymine all together how much percent? 30%. Total 100%. Total 100% na? Minus 30%. Equal to 70%. 70% have two party. 35% what? Cytosine. 35% guanine. So first question came. Number of nucleotide of guanine. I am changing the question. Second question. Number of nucleotide of guanine. So first question answer done. Percentage of cytosine is how much? 35%. Answer is there. You understood or not understood? Tell me. Cytosine is 35%. Everybody understood? Someone wrote also. Someone wrote complete answer also. Well done. Very good. Now I am coming on the second question. Number of nucleotide of guanine. How many nucleotide of guanine will come? Let me solve. So how many? Guanine is how much percent? Guanine is how much percent? 35%. So what will be the number of nucleotide of guanine? So base pair is how much? 2000. Base pair equal to 2000. So nucleotide equal to 4000. Why 4000? Because in one base pair, 2 nucleotide is there. In one base pair, 2 nucleotide is there, na? Got the idea? So base pair is 4000 and guanine is how much percent? 35%. So 4000 into 35 upon 100. How much coming? 0 so 0 cancel, 0 so 0 cancel. 34 into 40. Multiply it. 35 4 ja 140 and another 10 will come. So how many nucleotide will come? 1400 nucleotide will come. 1400 nucleotide will come. Got the idea? Is it clear? Done? Over? Right. Well done. So second question is also done. Number of glycosidic bond. I am telling you a formula for glycosidic bond. Number of glycosidic bond equal to two n minus 2, where n equal to base pair. So how many base pair is there? How many base pair is there? 2000 base pair is there. So 2 into 2000 minus 2 is equal to glycosidic bond. Got the idea? What is the formula for glycosidic bond? 2n minus 2. 2n minus 2. Got the idea? So it is coming how much? 3998. Done? Okay, ji, done, over. So this is also done. Number of nucleosome. So you better know one nucleosome have how many base pair? One nucleosome 200 base pair. 200 base pair. In packaging of DNA you have studied about. So number of nucleosome, total how many base pair is there? So total base pair is 2000. So 2000 divided by 200. 0, 0, 0, 0 cancel. How many? 10 nucleosome. Got the idea? 10 nucleosome. The last question, number of hydrogen bond. Number of hydrogen bond. So number of hydrogen bond, we have to write. Let me erase this area. So adenine and thymine total percentage is 30. So adenine double bond thymine, it is how much percent? Together 30% of 2000. 2000 base pair is there, na? Right? Cytosine and guanine have triple bond. And cytosine, guanine all together how much percent? 70% of 2000. Now tell me what is the value of 30% of 2000? 600 base pair. And 70% of 2000. 1400 base pair. Let's check 2000 coming or not. 2000 coming means we are in right direction. Adenine, thymine have double hydrogen bond, multiply by 2. Cytosine, guanine have triple hydrogen bond, multiply by 3. 6 2s are 1200. 14 3s are 4200. Total 5400. Got the idea? Is it done? Is it clear? So these are the numerical related to Chargaff rule. You got the idea? Is it clear in the mind? Done, over. Now I am moving ahead. Now I am going to talk about from search of genetic material you keep 2-3 things. Scientist associated with transforming principle. Is Griffith. Got the idea? Another scientist, DNA is transforming material. It is told by which one? MacLeod, Carty, and Avery, and ETL. These are the scientists told DNA is transforming material. Unequivocal proof of DNA as genetic material. Scientist associated with it is what? Harsh and Chase. Got the idea? Anything you want to know in detail? Right. Now I am telling you DNA replication. DNA replication. It is a right semi-conservative replication. This semi-conservative replication given by Watson and Crick. Got the idea? So DNA replication is semi-conservative, semi-discontinuous, and bidirectional process. Bidirectional process. Enzyme associated with DNA replication: helicase. Opening of DNA helix. Primase. Initiation of DNA replication. Initiation of DNA replication. It is performed by primase by forming RNA primer, not DNA primer. I am not telling by mistake. Forming RNA primer. Many times students think, sir, have to write DNA primer, but they wrote mistakenly RNA primer. It is RNA primer. Now another enzyme is DNA dependent DNA polymerase. Polymerization of new strand. In which direction? Always in 5 to 3' direction. Ligase. Joining of DNA fragment. What is leading strand? Continuously synthesized new strand is called leading strand. What is lagging strand? Lagging strand. Discontinuous strand. First discovered by Japanese scientist Okazaki, and hence it is called Okazaki fragment. You got the idea? Now I am telling one question based on experiment of Michelson and Stahl. Got the idea? So experimental proof of semi-conservative replication. Experimental proof of semi-conservative replication. What you don't forget from here is, it is given by two different scientists. One, Michelson and Stahl. Another is Taylor. Michelson and Stahl work on E. coli. Used non-radioactive, mind this line, non-radioactive heavy nitrogen. Non-radioactive hydrogen, heavy nitrogen. So Taylor work on fava bean. It is Vicia fava and use radio level thymidine. Use radio level thymidine. You keep these things in mind. Got the idea? First, these experiments performed by Michelson and Stahl. Later in same year is done by which one? Taylor. One work on prokaryote, another work on eukaryote. Another one have taken heavy nitrogen, another have taken which one? Radio level thymidine. Clear? Now, I am just telling you the question how it is going to ask from here. How the question going to ask from here. So I am asking a question. It is better explain the things through a question and then I will explain it. If we place 20 bacteria containing heavy nitrogen that is N15. This is a bacteria. Such type of 20 bacteria we are taking heavy nitrogen in a culture medium containing N14H4Cl. N14 as source of nitrogen. Then after 60 minutes, how many bacteria contain DNA of light density? Suppose that this is the question going to ask in NEET, how you will handle it. So first let me tell you the meaning of heavy, medium and intermediate. My intention is to explain this question. If such type of question came in NEET, you don't make a mistake. Just have a look. So if both DNA have 15-15, it is called heavy. If one strand is 15, another strand is 14, it is called medium or intermediate density. And if both DNA is what basically 14-14, it is called light density. Light strand or light density strand. You got the heavy, medium and light. Now in 60 minutes what happened? So first you better know bacteria, and I am writing especially the name also, E. coli. Bacteria, and especially which bacteria? E. coli. One bacteria become two bacteria. You better know generation time of the E. coli bacteria is 20 minutes. So in 20 minutes, one bacteria converted into two bacteria. So we have how many minutes? 60 minutes. So let's check. Got the idea? So you got the meaning of heavy, medium and light. Now I am erasing this side because I want to write something here. So first see with one bacteria, then in last we will talk about 20 bacteria. So I am taking a one bacteria with heavy DNA. This is heavy DNA bacteria. One bacteria have how many DNA always? One DNA. So this is one bacteria have heavy DNA, but we are growing in N14 medium. So DNA replication occurs semi-conservative, so old strand will separate. It is N15, N15. But what the new strand will came is 14. It is after 20 minutes. First generation. Both bacteria become medium density. Agree or not? Because both bacteria have one strand N15, another strand N14. Now after another 20 minutes, 2 bacteria convert into 4 bacteria. So separate it first, old strand 15, old strand 14, old, I am writing, old strand 14, old strand 15, agree or not? Old. Now I am adding new strand. New is always 14 because source is 14. So here 14, here 14, here 14, 14, here 14. So this bacteria 14 means light, this bacteria light, this 14-15 medium, this 14-15 medium. Did you got the idea? Now after another 20 minutes, it is 40 minutes, not 20 minutes plus 20 minutes, 40 minutes. Now another 20 minutes, 4 bacteria become 8 bacteria. Now I am telling you a tricks. 1 bacteria converted into 2 bacteria, 2 bacteria converted into 4 bacteria, 4 bacteria converted into how many bacteria? 8 bacteria. Now you keep in mind, first and last have medium, 15, 14. First and last have medium, always it remain. Always, because 115 will go na, so first and last here see medium. Remaining how many DNA is there? 6. 1, 2, 3, 4, 5, 6. These all become which one? Light. Light. So how many bacteria have light DNA? How many bacteria have medium density? So 6 bacteria, 6 by 8 is which one? Light. 6 by 8 bacteria is have light DNA, and 2 by 8 bacteria have medium DNA. Agree with me or not? First you tell me, are you agree with me or not? I am writing 6 by 8 bacteria have light DNA, 6 by 8 bacteria have light DNA, and 2 by 8 bacteria have medium DNA. Now how many bacteria we have? 20 bacteria we added. We not added 1 bacteria. So 20 bacteria become how many bacteria? In one generation after 20 minutes, 40 bacteria. Second generation after another 20 minutes, 80 bacteria. Third generation, it is 60 minutes, na? 20 minutes, 20 minutes, 20 minutes, 160 bacteria. So we are here 160 bacteria is there. And out of 160 bacteria, how much are light? I am asking light, na? 6 by 8 are light. So 160 into 6 by 8. 8 2 are 16. 20. How much? 6 2 are 12. 120 bacteria. 120 bacteria have which one? Light DNA. 120 bacteria have light DNA. You got the answer? You got the answer? Is it clear? Is it clear? Done? Understood? Picture came. First you do with the 1 bacteria. So simply you have to keep in mind what? Heavy bacteria, 15. Anybody asking after how many generation? 4 generation. Or asking after 80 minutes. So after 80 minutes, 1 bacteria converted into how much? After 80 minutes, 1 bacteria converted into how much? 1 to 2, 2 to 4, 4 to 8, 8 to 16. 20, 80 minutes. 16 bacteria. So, or 16 DNA. So out of 16 DNA, only 2 will be how much? Always 2/16 is medium, 4/16 is normal. Sorry, normal, huh? Normal. Sorry, 14/16, nah? 14/16 is normal. If it is how many generation they are asking? For 80 minutes. So likewise, only 2 remain medium, remaining become which one? So in this question, 60 minutes are there. So 2/16 medium. Sorry, 2/8, na? 60 minutes. So 1 minute, 1 bacteria to 2 bacteria, 2 bacteria to 4 bacteria, 4 bacteria to 8 bacteria, 20, 20, 20. Out of 8 bacteria, 2/8 medium, 6/8 normal or light. I am asking light, 6/8. Total bacteria, how much came? So I have given you 20 bacteria, 20 to 40, 40 to 80, 80 to 160 bacteria. Out of 160, I did what? 160 into 6/8. Answer came 120. Done? Is it clear? Now we are moving ahead. So these are the questions which is going to ask from which one? Michelson and Estall type of experiment. Now I am telling you another very important area from where question is going to ask. This is a DNA. This is promoter. This is terminator. This is a structural gene. Suppose that here A, T, C, G, T, A is there. So in this T, A, G, C, A, T is there. This is a structural gene. Got the idea? 5 prime, 3 prime, 3 prime, 5 prime. So 5 prime to 3 prime strand is called 5 prime to 3 prime strand is called which one? Coding strand. Or it have a other name, sense strand. So whoever 5 prime to 3 prime, suppose that I am telling this strand is a coding strand. Means this is 5 prime to 3 prime. So both coding strand and promoter act as a reference. Here promoter is there. So near the promoter here 5 is written. So we will read it 5 prime. Near the promoter here it is written 3. So we will read it 3 prime. So here 5 prime to 3 prime. Here 3 prime to 5 prime. So coding strand, and which strand? 3 prime to 5 prime strand is also called template strand. Template strand, or it is also called sense strand. Sorry, anti-sense strand. Anti-sense strand. What you have to keep in your mind is during transcription, RNA is like coding strand, and you are habituated with mRNA. So I am writing here like that. mRNA is like coding strand and complementary to template strand. Try to understand with an example. I am asking a question. If DNA have 5 prime A, T, C, G, T, A, T, T, 3 prime. Then in RNA, mRNA, what will be the sequence? Tell me. Then in mRNA, what will be the sequence? Tell me. DNA I wrote 5 prime A T C G T A T T 3 prime. Then in RNA, what will come? So I told here mRNA is like coding strand. So who is coding strand? 5 prime to 3 prime. Got the idea? Who is coding strand? 5 prime to 3 prime. So mRNA will be what? 5 prime. A in RNA, who is not there? T. In place of T, who will come? U. C. G. U. A. U. U. 3 prime. So this mRNA prime is also same. It is 5 prime to 3 prime. It is also 5 prime to 3 prime. You got the idea? You got the idea? Is it clear in the mind? So mRNA is like coding strand. I am asking another question. If DNA have 3 prime, A, C, G, T, 5 prime in DNA, then after transcription in mRNA, who will come? After transcription in mRNA, who will come? So it is which strand? 3 prime to 5 prime. It is template strand. Got the idea? So I wrote mRNA is like coding strand and complementary to template strand. Complementary. So here 3 prime to 5 prime, it becomes 5 prime, A pair with U, C pair with G. It is called complementary. G is complementary to C, T is complementary to A, and it becomes 3 prime. So how to handle the question? You got the idea? Understand? Clarity came? Can I give you another question? Okay, Ji. Okay. Another question, you solve it. DNA 5 prime C G G C A C 3 prime. Then in mRNA, right? Prime also and sequence also. Prime also and sequence also. So it is same. In mRNA 5 prime C G G C A C 3 prime, because it is which strand? Coding strand. mRNA is like coding strand. If I change the prime, if I wrote in option in DNA 3 prime C G G C A C 5 prime DNA, then in RNA what happened? It become opposite. It is template strand not is there. It is template strand is there. So what happened? It become 5 prime. C pair with G, G pair with C, G pair with C, C pair with G, A pair with U, C pair with G, 3 prime. You got the idea? How to handle the question from this transcriptional unit? Is it done? Now I am telling you exon, intron. This term people can also ask. Exon is which one? Coding sequence of DNA. Its information is present in processed RNA, processed mRNA or mature mRNA. Intron is what? Non-coding sequence. Its information present in, not present in processed RNA. Not present in processed RNA. These exon intron present in eukaryotic gene. Eukaryotic gene. In eukaryote, which gene is present? A split gene is present. In eukaryote, a split gene is present. A split gene means what? Exon is interrupted by intron. Got the idea? Now I am telling another information examiner ask. Polycistronic mRNA. These line also going to ask in exam. When an mRNA have information form more than one cistron. Polycistronic mRNA or polycistronic gene present in which one? Prokaryote. Eukaryote is usually monocistronic. Usually monocistronic. Eukaryote is usually monocistronic. Now, prokaryotic transcription and translation. Where the location? Tell me. Prokaryotic transcription and translation occur in simultaneously in cytoplasm. So in prokaryotic transcription, 3 steps are there. Steps. First steps, initiation. It is done by when sigma factor join with RNA polymerase. In prokaryotic transcription only performed by RNA polymerase. And third, termination occur when Rho factor join with RNA polymerase. When Rho factor join with RNA polymerase, transcription occurs. So transcription in prokaryote is over. Simply initiation occur by which factor? Sigma factor, but not alone when it attach with RNA polymerase. Elongation occur when sigma factor detach and it performed by RNA polymerase only. Termination occur when Rho factor join with which one? RNA polymerase. So these are the prokaryotic transcription. Got the idea? This is about which one? Prokaryotic transcription. Got the idea? Is it done? So one tricks I am giving you. Suppose that someone saying this is a DNA and it is forming a new DNA. DNA to DNA. Which enzyme will use? Enzyme will use DNA dependent DNA polymerase. One DNA converting into 2 DNA with the help of which enzyme? DNA dependent DNA polymerase. Suppose that here it is a DNA and here who is forming mRNA. Which enzyme will use? DNA dependent. It depends on DNA. But who is forming? RNA. So enzyme become RNA polymerase. Got the idea? Suppose that here it is RNA is there, mRNA is there, and from it DNA is forming. It occur in retrovirus, na? This DNA is called cDNA. When DNA form from RNA, this is called cDNA, complementary DNA. Here which RNA is forming? Which enzyme is acting? Who is act as a template? mRNA. So we will say RNA dependent. DNA polymerase. Got the idea? Another information is every polymerase, any kind of polymerase, perform polymerization in 5' to 3' direction. So any new strand who are forming always form in which direction? 5' to 3' direction. Got the idea? Any, either RNA is forming or RNA to DNA is forming or DNA to DNA is forming. Any time the new strand when forming is forming in which direction? 5' to 3' direction. So polymerization always occur in 5' to 3' direction. You got the idea? So DNA to DNA formation done by which enzyme? DNA dependent DNA polymerase. DNA to RNA formation occur by which enzyme? DNA dependent RNA polymerase. And mRNA to cDNA. When mRNA making a DNA, that DNA is called cDNA. That formed by which enzyme? RNA dependent DNA polymerase. Why RNA dependent? Because they are dependent on mRNA and making DNA. So RNA dependent DNA polymerase. Is it clear? Is it all right? Done? Okay, ji. Now we are moving ahead. Now just give me a 1 minute break, water break. You also take a water break. Then I am moving ahead. हाँ जी, see this point also. This is important. Central dogma of protein synthesis. It is given by Crick. Right? Central dogma protein synthesis given by Crick. What happened? It is saying about flow of genetic information. How information flow occur? From DNA information go in RNA. From RNA information flow in protein. So this is the direction of flow of the genetic information, DNA to mRNA to protein. So formation of DNA to DNA is called replication. Formation of mRNA from DNA is called transcription. Formation of protein from mRNA is called translation. In some retrovirus, mRNA convert into DNA, and this event is called reverse transcription. These occur in some retrovirus, example HIV. Not E, just a minute. Human immunodeficiency virus. Reverse transcription occur. Now move ahead. These are the area also questions possibly going to ask. That is property of a genetic material. These are the point which is important from property of genetic material. Any kind of genetic material must contain these properties. So first, it have power to make it replica. Only who have power of replication act as genetic material. So first property of genetic material is replication. This replication present in both DNA and RNA. DNA and RNA both have power of replication. So based on first property, both DNA and RNA act as a genetic material. Second property, it should be chemically and structurally stable. Who is stable? DNA. Who is not stable? RNA. Why RNA is not stable like DNA? Many reason is there. RNA act as a catalyst. Who are catalysts are reactive. That is one of the reason RNA is not stable. RNA have hydroxy group on second position of ribose sugar. That is highly reactive. Who is more reactive is less stable. And in RNA, uracil is there in place of thymine. That is the reason RNA is less stable than DNA. DNA don't have deoxyribose sugar. DNA not act as a catalyst. DNA have thymine in place of uracil. That make DNA as better genetic material than RNA because DNA ensure more stability to the genetic material. Third property of the genetic material, it should provide a scope of slow change. Slow mutation, not fast mutation. And this slow mutation property present in which one? DNA. RNA is fast mutating. So based on second and third property, who is serving as a better genetic material? DNA. Now fourth property of the genetic material is it able to express itself to form a Mendelian character. Form a Mendelian character means expression of gene, formation of protein. Able to express itself as Mendelian character means able to form a protein, and that protein act as enzyme and enzyme control a character. So who can express better? RNA. Because for making protein, RNA only go for translation, but DNA depend on the RNA for protein synthesis. So who express better itself as Mendelian character? RNA. But Based on stability or scope of slow changes, DNA is serving as a better genetic material than RNA. Hence, in evolution process, first who came on earth? RNA came. NCERT told it is RNA world. Beside of RNA came first on planet, but RNA restricted as a genetic material in few viruses only, and in most of the organism who evolve as a genetic material, DNA evolve as a genetic material, because DNA is a better genetic material over RNA. How better genetic material over RNA? Because it is stable and there is a scope of slow changes. So this paragraph is also important to keep in the mind. Now we are moving ahead. From here, one data is important, very basic thing, but I want to say because directly question is going to ask. That is the reason. My intention is not here right now what you know, what you don't know. I have only intention from where important questions is going to arise. So NCERT wrote कि in prokaryote, only one type of RNA polymerase is responsible for formation of all three type of RNA. tRNA, rRNA, mRNA in prokaryotes synthesized by how many different enzymes? Only one. That is DNA dependent RNA polymerase. But in eukaryote, there are different enzymes are there for formation of different type of RNA, like RNA polymerase 1 transcribe ribosomal RNA. Don't forget. Don't forget this line. It may be in exam, it directly came in a question like match of the following. Ribosomal, RNA, rRNA produced by which RNA polymerase in eukaryote? RNA polymerase 1. Next is what? Next stage, here you keep in mind, which type of ribosomal RNA in eukaryote produced by RNA polymerase 1? So these are the ribosomal RNA, 28S, 18S and 5.8S. Now, RNA polymerase is responsible for, RNA polymerase 3 is responsible for formation of which one? tRNA, and 5S rRNA and snRNA. So tRNA, 5S rRNA and snRNA, small nuclear RNA produced by which RNA polymerase 3? Don't forget this line. And RNA polymerase 2 is responsible for transcription of precursor of mRNA, that is called heterogeneous nuclear RNA, hnRNA. These are the data which is important for exam. Please don't forget it. Got the idea? Next we are moving on genetic code. What you don't forget from here is first the name of a scientist, that is what? Gamo. Where Gamo is written here? George Gamo. George Gamo was a physicist and advocate about what? Ki genetic code are triplet. Genetic code are triplet, that is advocated by a physicist. The name of the scientist is what? George Gamo. Got the idea? Now another scientist name they will ask. Chemical method developed by whom? Hargobind Khorana. Who developed the chemical method? Hargobind Khorana. Who told genetic code are triplet? George Gamo. Now another scientist name you don't forget from here is polynucleotide phosphorylase. It is popularly called Severo-Ochoa enzyme. These enzymes are helpful in polymerizing RNA with definite sequence in a template-independent manner. Right? Enzymatic synthesis of RNA, the enzyme who is responsible for this, right, is which one? Polynucleotide phosphorylase. It is also called Severo-Ochoa enzyme. It performs polymerization in template-independent manner. Another scientist you don't forget here is Marshall Nierenberg. He developed cell-free system for protein synthesis. So, cell-free system of protein synthesis developed by Marshall Nierenberg. Enzymatic synthesis of RNA that done by Severo-Ochoa enzyme, that is which one? Polynucleotide phosphorylase. Chemical method developed by which one? Hergovin Khorana, that make homo and copolymer. Copolymer is heteropolymer. Homopolymer and copolymer. These three scientists you don't forget. It is important for the exam. Now we are moving ahead on property of the genetic code. So if we talk about genetic code, have these property, you don't forget it. First genetic code is triplet. Got the idea? Another genetic code is contiguous. Contiguous means commaless. No sign of punctuation is there. Genetic code are non-overlapping. Another property of genetic code are nearly universal. Another property of genetic code is degeneracy. And another property of genetic code is unambiguous. So these are the some of the property of genetic code. So I hope you know the meaning of most of, I am telling the meaning of few. You know triplet. 3 nitrogenous base make a code like UUU is a genetic code. It code for which amino acid? UUU code for which amino acid? Tell me fast. Phenylalanine. Contiguous means no sign of punctuation between two code. Here AUG is written, in between AUG and UUU, no comma, no full stop, no bracket is there. So it is contiguous. Genetic code is non-overlapping. Non-overlapping means this is one code, this is another code, this is another code. AUG, you cannot say, it is correct method, you cannot say, look sir, you want triplet na, it is called overlapping, it is wrong. AUG came in first triplet also and UGU also. It is wrong, it is right. It not overlap. Got the idea? It is nearly universal means if UUU is a code, it code for same amino acid, that is phenylalanine, in every species. Some exceptions are there, but in bacterial and in mitochondrial and protozoan genes some exceptions are there. Otherwise, meaning of a code is same in every species. UUU code for phenylalanine in human also, code for phenylalanine in bacteria also. Based on this property of genetic code, genetic engineering is based. You are adding a human gene in bacteria and the same gene producing same protein in bacteria also, it is possible due to this property of genetic code. Genetic code are nearly universal. Unambiguous means a particular code for only one amino acid. UUU only code for phenylalanine, no any other amino acid. UUU only code for phenylalanine, no other amino acid. This is the meaning of which one? Unambiguous. Degenerate means there are more than one code for one amino acid, like GUG, GUU, GUC, GUA. I wrote 4 code. These all are responsible for same amino acid valine. So this is one amino acid are coded by more than one code. This is called degeneracy. This is called degeneracy. I hope you understand the meaning of every one. Now NCERT asks very beautiful question here. We are going to discuss this question also from the genetic code. NCERT asks question here. They have given you, wait a minute. What happening? NCERT have given, see there, they are asking a very beautiful question here, you must know it. A start and a stop codon everybody knows, so I'm not telling. AUG is the start codon and UAA, UAG, UGA is the stop codon. Now look at this question. If following is the sequence of nucleotide in mRNA, predict the sequence of amino acid coded by it. AUG is given. Can you say the amino acid? Yes, I can say. Which amino acid? Methionine will come, because AUG code for methionine. If UUU is given, can you say? Yes, I can say phenylalanine will come, because UUU only code for phenylalanine. If UUC is given, can you say? Yes, I can say. Who will come? Phenylalanine will come. Once again, UUC is there, phenylalanine. UUU is there, phenylalanine. UUU is there, phenylalanine. UUC is there, phenylalanine. So if we know the mRNA sequence, we can say easily about the amino acid sequence. Agree or not? Agree or not? If you know the mRNA, you can say about the amino acid or not? Yes. NCERT asks, it is possible due to which property of genetic code? Tell me. We can predict the amino acid sequence from mRNA sequence. It is possible due to which property of genetic code? Answer it. Answer it. Which property of genetic code is there? Non-ambiguous. Well done. Non-ambiguous is there. Because we know UUU not code anything other than phenylalanine. AUG not code for any other thing other than non-ambiguous, means no confusion in meaning. If one word have two meaning, confusion came na. Here one word have only one meaning. UUU means phenylalanine. UUC also means phenylalanine. AUG means methionine. Got the idea? Now NCERT asks a question. Predict the nucleotide sequence in the RNA from methionine, phenylalanine, 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 phenyl. Can you do it? Can you tell about mRNA from amino acid? Can you tell about amino acid to mRNA? It is not possible. Why not possible? Phenylalanine is there. How can I write the mRNA? It may be UUU, it may be UUC. How can we decide it is UUU or UUC? We cannot decide, na? We cannot decide. So we cannot say mRNA from protein, and it is due to which property of genetic code? Degeneracy of the genetic code. Degeneracy of the genetic code. Got the idea? Understand? So this is about the genetic code. Now we are going to talk about some basic idea about mutation. See, gene mutation. Gene mutation is also called point mutation. No need to get confused. Point mutation or gene mutation are same thing. It is of two types. right? One is substitution mutation. Another is frame shift mutation. Substitution mutation and frame shift mutation. What happened? Substitution mutation is of two types: transition and transversion. In transition, what happened? Purine replaced by purine. Or pyrimidine by pyrimidine. Here, purine by pyrimidine or vice versa, pyrimidine by purine. So such kind of mutation of replacement, suppose that in a DNA here A is there, in place of A T is coming. Which kind of mutation? A is substituted by replaced by T. Which type of mutation will you say? In place of A T is coming. Frame shift mutation or substitution mutation? Substitution mutation. Which type of substitution mutation? Transition or transversion? What kind of substitution mutation? Transition or transversion? What you will say? A replaced by T. Adenine is a purine. Thymine is example of pyrimidine. In place of purine, who is coming? Pyrimidine is coming. So it will come in what one? Transversion. Purine is replaced by pyrimidine. Purine by pyrimidine or pyrimidine by purine. It come under which one? Transversion. Got the idea? Is it clear in the mind? Is it all right? Now I am talking about frame shift mutation. It is of two types: addition and deletion. And NCERT explain it with the help of a beautiful example. So I am taking NCERT example itself only. If anybody asking you a question, you can take the help of your NCERT and you can answer it. Sickle cell anemia is due to which kind of point mutation? Sickle cell anemia is due to which type of point mutation? Answer it. I am giving you some break and find the answer of the question. Somehow you are also on screens continuously. So in finding the question, certain mental deviation will occur. Option I will not give. Answer without option. Transversion. Very good. Transversion. You have seen, na? GAG become which one? GUG. Got the idea? Is it? In mRNA, what the idea? In DNA, A replaced by which one? T. In DNA. So it is which kind of mutation? Transversion. Got the idea? Is it clear? Done? So sickle cell anemia is example of transversion. Okay, now we are going to talk about which kind of mutation? Frame shift mutation. Look here. So suppose that this is a triplet codon. So 1, 2, 3, 4 codon are there. Ram has red cap. Now we did addition. We add which one? B. So when we are adding B, so after addition, sequence change. Before addition, change not occur. Ram remain Ram, Haj remain Ram, Haj. But what happened? Red become BRE and cap become DCA. So sequence gets changed. reading sequence is going to change. So when codon will change, amino acid will different, na? So entire amino acid during protein synthesis and such type of mutation came different after the point of additions of the new nucleotide. Now we are adding two nitrogenous base, B and I. Then also change occur. Ram remain Ram, Haj remain Haj. Before mutation, no problem. After, red become BIR, cap become EDC. Here also frame shift occur. Reading frame entirely change. Now when we are adding the three nitrogenous base, so Ram remain Ram, Haj remain Haj, red remain red, cap remain cap. No frame shift occur. Only a new amino acid added. So frame shift mutation occur due to addition or deletion of one or two nitrogenous base. When you are adding or deleting three or its multiple of nitrogenous base, frame shift not occur. So this is about the mutation, that is also over. Now we are coming on which one? Protein synthesis. You want to know about protein synthesis? Nothing like it is a concept. So in I think revision it will not going to help you much. Simply in protein synthesis you keep in mind here is what I am telling you. So you just talk about in protein synthesis tRNA, you just inverted L-shape structure present in which one? tRNA. Actual structure is 3D structure, it is inverted L shape. 2D structure of tRNA look like which one? Clover leaf. Got the idea? Amino acid attached at which site of the tRNA? 3'. Anticodon arm of the tRNA pair with which one? Codon arm. Getting the idea or not? Because mechanism is there, I can't say factual nature of question from here. And if I start teaching it, a problem arises what? Entire time will go in this only. So here I am telling lac operon in very fast. And I will draw the diagram, then I will say. So see, I am just telling you about lac operon in E. coli very fastly. Because we have to move on many other chapter. We have expended a lot of time in this chapter. Okay. Lac operon on E. coli. In E. coli. Oh. So you just see without paraphernalia I am saying it, no time, too much introduction. Have a look here. So here it is promoter for regulator gene. Here it is regulator gene represented by I. Here it is promoter for structural gene. It is operator and this is a structural gene ZYA. Got the idea? So this regulator gene is a constitutive gene or housekeeping gene. Constitutive gene work all the time and a structural gene is a luxury gene. It work on demand. So what happened? First of all, transcription of regulator gene occur. So this promoter is there, RNA polymerase bind here and it is going to form mRNA. And this mRNA undergoes in translation. And as a result, it forms a protein. This protein is called active repressor protein. Active repressor protein. And these active repressor protein bind at operator. Due to binding at operator, partially promoter of a structural gene become hidden. And this RNA polymerase for a structural gene not able to bind at promoter for a structural gene because it partially hide the structural gene also when it bind at operator site. So due to that reason, when RNA polymerase not bind, this structural gene undergoes no transcription. And if mRNA will not form, no translation. So these regulator gene is responsible for switching off condition of. So who is in switch off condition? A structural gene is in switch off condition. A structural gene in switch off condition because these regulators in making active repressor protein binding of operator. Due to that reason, promoter region not get bind by RNA polymerase, no transcription, no translation, no protein synthesis by a structural gene. These structural gene are switch off. Got the idea? Now what happened when the twist came in the story, when lactose enter in the environment of cell? When who enter in the environment? Lactose enter in the cell environment. Lactose is which one? Beta galactoside. It is a sugar. It is a disaccharide. So these lactose when present in the bacterial cell environment, absorbed by bacteria, these bind with the active repressor protein and make it inactive. As it become inactive, what happened? They are not able to bind at which side? Operator side. Now who is visible? Promoter is visible. RNA polymerase for a structural gene bind. Transcription occur. Are you getting the idea? And when transcription occur, mRNA is going to form. It is polycistronic mRNA because it have information of 3 cistron. Got the idea? And then Z gene producing which one? Beta galactosidase. Y is in forming enzyme permease. And A is in forming which one? Transacetylase. Got the idea? So it becomes switch on. So as long as lactose is there, these structural gene remain switch on. So lactose act as inducer. And hence lactoperon is called inducible operon. And it is under negative regulation by a structural gene. So keep something in mind. Why these enzymes are producing in presence of lactose? Because all enzymes are associated with lactose metabolism. If lactose is not there, what beta galactosidase will do? Nothing. Beta galactosidase is a lactose digesting enzyme. Mind it. It is important line. Beta galactosidase digests the lactose. Permease is responsible for absorption of lactose. So these all enzymes is associated with lactose metabolism. So when these lactose coming, then the need of these enzymes are there. And in that case, this gene become active and forming this enzyme. In absence of lactose, there is no need of these enzymes are there. NCERT wrote 2 more lines, don't forget it. A very low amount of lac operons are open, working all the time. Another thing, lac operon is under both positive and negative regulation. At this stage, you studied negative regulation. What is the positive regulation you will study in the higher classes? Now saying bye-bye to this chapter. Now we are coming on another chapter. In Mendelian genetics, you say, what should I say? Because their numerical type of things are more. So what you will say, I will give you a brief synopsis of the Mendelian genetics. Tell me, I am working here based on you, where you are stucking. Gamete formation, everybody know, na? Every student, one question probably going to ask from gamete formations. Got the idea? Numerical type of thing. Okay. See, first I am saying you gamete formation. I hope everybody know it. It is a high time. So gamete formation, suppose that anybody asking you capital A, small a, capital B, small b, capital C, how many type of gamete produce? So types of gamete, there is a formula. equal to 2 is to the power n. Everybody know probably where n equal to number of heterozygous character. You got the idea? Right? Now if we talk here, how many heterozygous character are there? Capital A, small a. Character factor occur in pair. So 1 pair, 2 pair, 3 pair. When both alphabet of same type, homozygous. When different type, heterozygous. So capital A, small a. It is different now, one capital letter, another small letter. So it is one heterozygous. Capital B, small b, heterozygous. Capital C, not heterozygous, homozygous. So how many heterozygous character are there? 2. So formula is 2 is to the power 2 equal to 4. What the idea? If anybody going to ask you a question, capital A, small b, capital C, how many gamete produced by it? Tell me. Capital A, small b, capital C, how many gamete produced by it? Answer it fast. So heterozygous character, how many? 0 heterozygous. All are homozygous characters. So 2 is to the power 0 equal to 2 is to the power 0 equal to 1. So one type of gamete came. So type of gamete over. Got the idea? Now I am telling you about, suppose that someone going to ask you a question, capital R, capital Y, small y, cross with small r, small y. What will be the phenotypic ratio? How fast you can say? What will be the phenotypic ratio? So here phenotypic ratio is 1:1:1:1:1 because it is a dihybrid test cross. See, this one is a test cross. Ratio is 1:1. This one is also a test cross. Here also 1:1. So 1:1 into 1. What happened? 1:1:1:1. Got the idea? Is it clear in the mind? Is it clear in the mind? Right? Now, I am asking another question. Capital A, small b, cross with small a, capital B, small b. Tell me what is the phenotypic ratio? Second question. I am coming with NCERT, it become easy, otherwise numerical don't have a limitation. Here, this cross with this, test cross, monohybrid 1:1. Here this cross with this, test cross 1:1. Once again a test cross. Now come on the agenda. I am just telling you about mono, I am writing some ratio, don't forget. Hybrid phenotypic ratio in F2 generation 3:1. Monohybrid genotypic ratio in F2 generation 1:2:1. Simply I am writing ratio. Now monohybrid ratio in which one? Codominance. Codominance ratio, phenotypic ratio. Phenotypic ratio in F2 generation 1:1. Codominant genotypic ratio 1:2:1. Incomplete dominance phenotypic ratio. Tell me, phenotypic ratio 1:2:1. Incomplete dominant genotypic ratio 1:2:1. Got the idea? Now, during incomplete dominance, if anyone you need explanation, I will explain anyone. F1 resemble with neither of the parent. Neither of the parent. Codominance. F1 resemble with both parent. Both parent. In dominance, F1 resemble with either of the parent. These are the lines sometime going to ask in exam. These are the lines sometime going to ask in exam. During incomplete dominant, F1 resemble with either of parent. Codominant, F1 resemble with both parent. And in complete dominant, F1 resemble with neither of the parent. Now I am asking a question. Capital A, small a, capital B, capital V. Wait a minute. Capital A, capital B, small b, capital C, capital small d, cross with capital A, capital B, small b, capital C, small d, small d. Tell me the phenotypic ratio fast. 30-second phenotypic ratio. So it is, I am asking a question. It is a monohybrid, dihybrid, trihybrid or tetrahybrid cross. Which cross? It is a monohybrid, dihybrid, trihybrid or tetrahybrid cross. Hybrid means heterozygous. Hybrid means heterozygous, na? So how many heterozygous is there? What ratio will come? Phenotypic ratio 3:1. Over. Here 4 character is there, but heterozygous character is only 1. If I ask a question, this cross with this, what answer you will give? Sir, phenotypic ratio 3:1. Got the idea? How? How many gamete produced by this? 2 type of gamete. How many gamete produced by this? 2 type of gamete. Punnett square form how many? 4. Which ratio will come? 3:1. Now I am telling you a question which will help you to handle many questions. I am asking a question, capital A, small a, capital B, capital V, capital C, capital D, small d, capital T, small e, cross with small a, capital B, small b, capital C, okay, capital D, small e. When we make a cross between these two, in what fraction B got small a, capital B, small b, capital C, small d, capital E, small e in their progeny? It is very easy question. In Mendelian genetics, you have to break everything in monohybrid. So cross this one with this one and tell me chances of getting this one. You tell me, I will write. Cross this one with this one and tell me the chances of getting this one. What will come? You make a cross. If you don't know, capital A, small a, cross with a small a. Two type of gamete, capital A, small a, here small a. Here who will come? Capital A, small a, here small a, small a. So chances of getting this one is what? How much? 1 by 2. 2 out of 1, na? Chances of getting capital B, small b out of this? Also 1 by 2. Chances of getting capital C, small c, capital C, capital C, capital C, small c, also 1 by 2. Capital D, small d, capital D, small d, chances of getting this one is 1 by 4. Chances of getting which one? Capital E, small e, small e, small e, and getting which one? Capital A, small e is which one? 1 by 2. Multiply it, 1 by 2, 2 ja 4, 2 ja 8, 4 ja 32, 2 ja 64, 1 by 64 answer. Okay. So break everything in the monohybrid. Is it all right? Now I am coming through the NCERT because numerical have endless job. So I think it is not wise to talk this time to numerical. We are just moving on the theoretical aspects. So give your attention. Where is Mendelian principle of inheritance? Keep these data in mind. Who is dominant, who is recessive? From here question is going to ask. See, test cross is important to know, I am telling you. Suppose that this is a violet flower and I want to know the genotype of this violet flower. Violet is a dominant trait. Okay. So violet may be capital W, it may be capital W, small w. Both will give which color? Violet color only. How to determine the genotype? Either it is capital W or capital W, small w. So for this purpose, we cross it with white flower pea plant, small w. If it is capital W violet, and when we cross with the white one, we got all violet progeny. So if we were crossing a violet with white, and all progeny are violet, 100% violet, you can say, कि sir, the violet given by you was capital W. Suppose that you are crossing a violet with white flower plant and you are getting in progeny 50% violet, 50% white in ratio of 1:1. You can say, sir, in that case, the violet given by you is which one? Heterozygous violet. So test cross, they help us to determine the genotype of dominant trait in F2 generation. Mendel used the test cross to determine the genotype. In F2 generation, he got 3 violet, 1 white. How can he say these 75% violet, who is homozygous violet? So he told na, कि violet coming in which ratio? 1:2 ratio. It is pure violet, it is heterozygous violet, and it is white. So these genotype he determined with the help of which cross? Test cross. Got the idea? Monohybrid test cross, what is the phenotypic ratio or genotypic ratio? In test cross, both genotypic and genotypic ratio are same. Monohybrid test cross, phenotypic ratio and genotypic ratio is 1:1. Dihybrid test cross, it is in 1:1:1:1. Trihybrid test cross, 8 times 1, 1:1:1:1:1:1:1:1:1. Check how many? 8 times 1. So number of 1 depend upon heterozygous. Trihybrid have 3 heterozygotes, so 8 type of gamete. How many gamete producing on the basis of that? So see, in test cross, genotypic and phenotypic ratio coming in which format? 1:1 format. Monohybrid 1:1, dihybrid 1:1:1:1:1, trihybrid 8 times 1. Are you getting the idea? So these are the test cross. It is helping us to determine the genotype. Now we are moving ahead. Give your attention. Haji. Law of dominance. From here people can ask the question. Mendel give 2 law based on the monohybrid cross. 2 law of inheritance based on monohybrid cross. One is law of dominance, another is law of segregation. Law of dominance is saying what? First thing is saying that character is controlled by discrete unit called factor. Discrete unit means who never mix. Another thing Mendel told, factor occur in pair in law of dominance. Another thing he told, in a dissimilar pair of factors, suppose that capital T1 T, a small t. So who express is called dominant, and another member who do not express is called which one? Recessive. So in dissimilar pair of factor, one member of the pair dominate the other. So here capital T is dominating over the small t because capital T, here it is becoming tall. So it is who is dominating is called dominant, who is not dominating is called recessive. Law of dominant also used to explain only one of the parental character in monohybrid cross in F1 and both in F2 in 3:1. So these are the outcome given by Mendel based on his law of dominance. Law of segregation, what told by the Mendel? In segregation, what happened? Here capital T small t, here capital T small t. Children is also small t small t. So these small t in the children only possible. These small t segregate from this capital T and these small t segregate from this capital T. Got the idea? So they are telling allele do not show any blending. They are present as separate unit, and that both the character are recovered as such in F2 generation due to which law? Law of segregation. Getting the things or not? If segregation is not there, these dwarf not going to express in the F2 generation. And based on dihybrid cross, Mendel have given the law of independent assortment. This is about example of which one? In complete dominance, they have work on which one? Snapdragon. Got the idea? Multiple allele based question I am saying. Something it is going to ask in exam. So let me say about multiple allelism. 5 minutes I will finish it. So come on, multiple allelism. I just want to write. Not just a minute. Just a minute. Multiple allele. Let me say in brief. When a gene has more than 2 different allele, it is called multiple allele. When a gene has more than 2 different allele, it is called multiple allele. It is reported in population. Listen it. Example, I gene. I gene have 3 different allele. IA has 3 different allele. IA, IV or IO or small I. But in an individual, 2 allele is present only. In an individual, maximum 2 allele of a gene is present. Maximum 2 allele of a gene is present. Got the idea? An example I am writing IA, another possibility IA, IO, another possibility IA, IB, another possibility IB, IB, another possibility IB, IO, another possibility IO, IO. So here IA is dominant over IO, IB is dominant over IO, and IA and IB are codominant. So here it is genotype. How many genotype you are finding for ABO blood grouping? 1, 2, 3, 4, 5, 6 genotype you are finding. And what will be the phenotype with this genotype? You will find this will give A blood group, this will give A blood group, A is dominant over O, AB are codominant, AB blood group, B blood group, B dominant over O, B blood group, here no one is there, O blood group. So how many different phenotype? 1, 2, 3, 4 different phenotype. Sometime examiner ask question based on number of genotype in multiple allele. No formula for phenotype. Number of genotype in multiple allele. So how to determine the number of genotype? Formula is n into n plus 1 upon 2, where n equal to different number of allele. N equal to number of different allele. Check the formula in this question also. In this question, how many allele are there? 3 allele. So 3 into 3 plus 1 upon 2. 3 into 4 upon 2. 2 2 is 4, 3 2 is 6 genotype came. How many 6 genotype is there or not there? Got the idea? Is it clear? So this is about the multiple allelism. Now I am going to tell you the question, one question based on blood grouping. Suppose that a female is there, have AB blood group. She is marrying with a male, have O blood group. What are the possible blood group in the children? So how to handle the formula? So I will write like IA, IB, IO, IO. Combination number 1, IA, IO. Combination number 2, IA, IO. Combination number 3, IB, IO. Combination number 4, IB, IO. So here A, here A, here B, here B. So possible blood group in children is A and B. We handle it like this. Now we are moving ahead. Actually, in genetics, what's happened? It is a conceptual area. So you cannot say in a short revision, very short revision to this. This requires comprehensive thing. Now I am telling about who rediscovered the Mendel. See, in fact, we are going to talk about. So in 1900, Mendelism is rediscovered by 3 scientists, De Vries, Correns and Sedmak. Got the idea? If someone asking you who study the during meiosis, observe that behavior of chromosome is like the behavior of Mendelian factor. That is done by which one? Sutton and Boveri, where the name is written. Got the idea? Sutton, Boveri, where the name is there? Not there? So chromosomal theory of inheritance is given by Sutton and Boveri. These are showing about the segregation of, it is showing what? A question we have given in a paper few days back and many of you make mistake here. So this is showing which one? G1 stage. 2 pair of chromosome is there. After S phase, doubling occur, formation of sister chromatid occur. During meiosis I at anaphase, mitosis, during mitosis as anaphase, who is segregating? Who is segregating? homologous chromosome. No, here who is segregating? At anaphase is there, na? Just a minute. Let me come on the complete diagram. It is meiosis. So here it is G1, it is showing already. Here after S, in G2 stage, see sister chromatase is there. During meiosis 1 at anaphase 1, who separate? Homologous chromosome separate. It is not centromere. Many times students did mistake metaphase. It is anaphase 1. Who is segregating? Homologous chromosome is segregating. During anaphase 2, sister chromatid is segregating. And this is the germ cell. These are the questions we have given for the labeling. Now, here in this question, it takes hour to explain, but I am telling in one line. A is proved as gene and chromosome. B is proved only as chromosome, not gene. Reason: one pair segregate independent of other pair, not always true for gene due to linkage. So gene can show linkage, but chromosome never show linkage. So out of A side or B side, it can prove chromosome, it also prove chromosome, it can prove gene, but it cannot prove gene. So if one liner answer, one is gene and this side is what? Chromosome. As according to NCERT, what the question they ask. Reason of third point, second side. Who can show linkage? Gene can show linkage, but chromosome never show linkage. Now the terminology. See, in which one? Marathon, we are finding maximum difficulty to dealing which one only? Genetics. Because here you cannot go for a shortcut. You cannot go for a shortcut. Ki do this line, it will come. And that is the reason we are finding maximum difficulty. I am finding maximum difficulty in the most interesting chapter. And the reason is this only. Ki you cannot go for shortcut here. Now, you just keep something in mind. From here, question is going to ask, theoretical question going to ask from here. Just a minute, give me a moment. This line is important. Morgan attribute this due to which one? Morgan find it in experiment, parental character is greater than recombinant character. And they told it is due to which one? Linkage. So anybody going to ask, term linkage and recombination is given by Morgan. Linkage and recombination is given by Morgan. These are the experiment of the Morgan. I am telling in 1 minute. So what done by Morgan in his experiment? He have taken Drosophila, a female Drosophila who have yellow body and white eye, crossed with wild type. Wild type means brown body and red eye. F1 generation he find crisscross inheritance occur. Crisscross inheritance means phenotype of father went in daughter and mother went in son. When he performed the mating between these progeny, in F2 generation he got 98.7% parental and 1.3% recombinant. In next experiment, he make a cross between white eye and miniature wing with wild type, red eye and long wing, male. F1, here also he got crisscross inheritance. Crisscross inheritance occur in the case of X-linked gene. Occur in the case of which one? X-linked gene. Later on, he make the mating of these two. He got in F2 generation 62.8% parental and 37.2% recombinant. And what conclusion he have given? He find in one case parental is more, 98.7, another it is 62.8 only. Why I am getting this type of result? So he told how much parental and how much recombinant will come, it depend upon distance between two genes on the chromosome. Both the genes are present on the X chromosome. So if distance between two genes are close, parental outcome is more. And if distance between two genes is more, parental outcome is relatively less. And based on that, he have given a conclusion. And what are the conclusion? He told linkage is inversely proportional to distance between two gene on a chromosome. Got the idea? And one of his student, Alfred Stewart Went, have developed the which one? Gene mapping. And what the formula we use during gene mapping? Simply I am writing the formula. Simply I am writing the formula for the gene mapping. And what is the formula for this? I am writing here recombinant frequency or crossover value is equal to total number of recombinant. It become a very boring chapter to deal in which one? Marathon. Very boring, most boring chapter. Divided by total number of progeny multiply by 100. Suppose that an example recombinant frequency came 5%. So distance between two genes is equal to percentage recombinant frequency. So 5% equal to 5 map unit. An example came 10%. So distance between two genes is 10 map unit, or we call centimorgan. Got the idea? Is it clear? Now come on another chapter. This chapter is not meant for marathon. Okay, ji? Now come on next. And next we are moving on cell cycle. Cell cycle. Cell cycle also a subchapter from where you can expect a lot of questions. Cell cycle. I am trying to revise this chapter in 20 minutes, because it is a very important chapter. You can expect 3, 4, 5 questions from here. Even 3, 4, 5 questions you can expect from here. That's the reason I am trying to devote 20-25 minutes on this particular chapter. Okay, ji? So let's start now. Okay. So who become most boring? Principle of inheritance. I just feel not comfortable to deal the important line from there, because all numerical are there. Na? So you cannot justify the numerical in that short span of 10 minutes, 20 minutes, 30 minutes. Okay, cell cycle. So see here, if we talk about cell cycle, I am dividing into two categories, interphase and M-phase. Got the idea? Interphase, if you talk about, we further classify into G1, S and G2 phase. 20 minutes I will try to fix. Let's see. If we talk about M phase, it have two Eve stages. One is karyokinesis, another is cytokinesis. Right, so karyokinesis is further subdivided into prophase, metaphase, anaphase, telophase in the case of mitosis, and in meiosis, prophase 1, metaphase 1, like, got the idea right now? So here G1 phase. G1 phase is also called gap 1 phase. It is also called post-mitotic phase, or it is also called pre-synthesis phase. S phase called synthesis phase. And G2 is called pre-mitotic phase or post-synthetic phase or gap 2 phase. Got the idea? Now we are going to talk about what important event occur in which phase. So if we talk about G1 phase, what important event occur? If we talk about G1 phase, what important event occur in G1 phase? So cell growth occur, RNA and protein synthesis, another thing organelle duplication. These events occur in G1 phase. Organelle duplication start. Got the idea? S phase, what happened? DNA replication. Or we can say chromosome duplication. Or you can write centriole duplication. And gap 2 phase, RNA and protein synthesis continue. Got the idea? Another thing, what happened in the gap 2 phase, cell organelle duplication completed. Got the idea? Now, I am telling you about S phase. It is very important. I will not say about all phase, don't worry. S phase, what important from where question is going to ask, I am going to say. S phase. During S phase, what happened? It is very important. During S phase, number of DNA, amount of DNA, number of chromatid, get double. Number of chromosome remains same. It is very important for numerical question. Have a look here. I am telling you with the example. It is important. You keep one thing in mind. One chromatid always have one DNA. One chromatid always have how many? How many chromatid is there? 2 chromatid. How many DNA is there? 2 DNA. How many chromosome is there? 2 chromosome. So chromosome 2. Chromatid 2. DNA 2. Agree? Everyone agree? Now what happening in S phase? Chromosome duplication. Formation of sister chromatid occur. That is called chromosome duplication. Now I am asking here a question. Chromosome how many? Chromatid how many? DNA how many? Tell me. So chromosome is 2. Chromatid is 4. DNA is 4. Number of chromosome always equal to number of centromere. So here 1 centromere, 2 centromere. Here 1 centromere, 2 centromere. So number of chromosome always equal to number of centromere. So 2 chromosome, 2 chromosome. 4 chromatid, 2 chromatid, 4 chromatid, 2 DNA, 4 DNA. Suppose that here amount of DNA is 2C. Amount of DNA, amount of 2 DNA is 2C. So amount of 4 DNA will be 4C. So can I write this thing? During S phase, number of DNA, amount of DNA, number of chromosome get double, number of chromatid get double, but number of chromosome remains same. This is justified or not justified? You understood? Understood or not understood? Clear? Now we are moving ahead. Okay, ji. Now give your attention. Now what to keep about prophase in mind? Prophase. Prophase you keep in mind, condensation of chromatin. And form chromosome. Another event of prophase is what? In interphase, interphasic nucleus, who was tangled? In interphase, sister chromatid or chromatin are tangled, get untangled in which stage? Prophase stage. Means earlier it was intercoiled, now it only coiled at the centromere. So it get untangled. What the idea? Shortening and thickening occur. 2 chromatid of a chromosome become visible. In late prophase, what happened? In late prophase, nuclear membrane, nucleolus, organelle like ER, Golgi disappear. This is about the prophase. Now we are talking about the metaphase stage. What is important for metaphase? You don't forget. Who is totally disintegrated, disappeared? Nuclear membrane. Now chromosome is spread in entire cytoplasm. Chromosome is spread in entire cytoplasm. The spindle fiber attached with the kinetochore. Got the idea? And all the chromosome arranged on a metaphasic plate, equatorial plate. That happened in the which one? Metaphase stage. Now what happened in the anaphase stage? Sister chromatids divide and move toward opposite pole. Sister chromatid divide and move toward opposite pole. This happened in the anaphase. Now I am asking a beautiful question here. Tell me the chromosome number in metaphase. In this cell, tell me the chromosome number. Chromosome number 2. Tell me the chromosome number in anaphase. 4, 1, 2, 3, 4. Each tetrachromatid is called a chromosome. So what important in anaphase? Centromere divide, sister chromatid move toward opposite pole, move toward opposite pole. Got the idea? And number of chromosome get double. Become double. Got the idea? Is it clear in the mind? Now if I ask a question, if G2 stage have, or G1 I am writing, G1 stage have 10 chromosome, number of chromosome at anaphase, at end of anaphase, what answer will you give? What answer will you give? 20. Because number of chromosomes get doubled at anaphase. Telophase is reverse of prophase. I am not telling much. I am just telling one numerical question, two numerical question. It will help you. Reverse of prophase. Now I am asking a question. If amount of DNA in G1 stage is 2C, then amount of DNA after. First question, S, G2, prophase, metaphase, anaphase, telophase, each daughter cell after cytokinesis. Answer it. Answer this question. G1 2C, then after S phase 4C, because amount of DNA double at S phase. G2 phase 4C, prophase 4C, metaphase 4C, anaphase 4C, telophase 4C, each daughter cell 2C. Got the idea? Each daughter cell 2C. Understood? Now another question. If the G1 stage, if at G1 stage number of chromosome is 20, then number of chromosome after. First option, S, G2, prophase, metaphase, anaphase, telophase, each daughter cell. So after S, 10. Number of chromosome not double during S phase. G2 10. Or sorry, 20 is there, na? After S 20, G2 20, in question 20 given, prophase 20, metaphase 20, anaphase double 40, telophase 40, each daughter cell 20. Parent cell 20, daughter cell 20, hence it is which division? Equational division. Got the idea? Done? So amount, keep one thing in mind, amount of DNA double in S phase, number of chromosome double in anaphase. Now meiosis. Meiosis 1 question is going to ask, so I am writing it. Prophase 1, I am not telling anything in meiosis other than prophase 1. Here question going to ask, and I am writing also, leptotin. Condensation of chromatin. You can see it is a question of need. Maximum chances one question will come from here. Maximum chances in 5 May, one question coming from what I am writing. So leptotin, condensation of chromatin start. Zygotin, pairing of homologous chromosome. pairing of homologous chromosome. Got the idea? In zygote, next what happened? This pairing of homologous chromosome is called synapsis. Formation of synaptominal complex. Another event is formation of bivalent and tetrad. Listen it carefully because it is a question, probably 90% chances is there a question will come. It is a matter of 4 marks, and if you did mistake, 5 marks. Bivalent or tetrad. These are the events you have to keep in mind occur during zygote. Okay. Now we are going to talk about pachytene. Crossing over or recombination occur. Between non-sister chromatid of homologous chromosome Got the idea? Another event occur in pachytene is what? Formation of, here everything is important, recombination nodule. Formation of recombination nodule, and crossing over is mediated by enzyme recombinase. It is mediated by enzyme recombinase. What the idea? So pachytene is also over. And another thing very important in pachytene itself, bivalent and tetrad clearly visible. Bivalent and tetrad clearly visible. This happen in pachytene also. Right? So pachytene, how many event is important? Listen it carefully. Crossing over or recombination occur first event. Crossing over always occur between non-sister chromatid of homologous chromosome. Is it clear? Another thing is what basically formation of recombination nodule, don't forget in pachytene, and bivalent and tetrad clearly visible in pachytene. It also not forget, please. You also not forget it, please. Got the idea? Bivalent and tetrad clearly visible. Now we are moving ahead. Give your attention. Now diplotin. Don't forget, once again it is a question of need. Diplotin. If we talk about diplotin, synaptominal complex dissolve. It is a protein complex. Homologous chromosome start to separate. Got the idea? Another thing is what? Formation of X-shape chiasmata occur. Formation of chiasmata. Got the idea? In certain vertebrate oocyte, remain suspended in this stage up to months and year. Okay, ji. Now we are going to talk about diakinesis. Terminalization of chiasmata. Chromosome get maximum condensation. Chromosome get maximum condensation. Spindle apparatus move on opposite pole and form a spindle. Got the idea? After that, nuclear membrane, nucleolus disappear. And it is called transition to metaphase. Transition to metaphase. Got the idea? This is about dikinesis. Okay, right? Okay. Complete disintegration of nuclear membrane marks the beginning of second stage. That is what? Metaphase. Now anything you want more in cell cycle, I will help you. If you are stuck in numerical, I can do something for you, but it will take 10 minutes. You decide. It will take 10 minutes. If you want to do something more with numerical, I can do more, but it will take at least 10 minutes. Then it will be better. Okay? You want? Or can we move on another chapter? Packet-in, of course, packet-in variation, responsible for variation. Okay, ji? So look here, I am just telling some numerical type of question, or I think in that way time we can do a lot of things. One explain picogram. Okay. Look. See here. This is a cell. It is going in meiosis I first. Forming two daughter cell. Actually, marathon is not for teaching the concept. It is for telling the important lines. But a teacher in us and a student in you. So we both came and stuck in which one? Something conceptual. It is not the right time to deal the concept. But anyhow, let's see, I am trying my best. Haji. So this is meiosis I, this is meiosis II. Look here. Now I am asking a question to you. And what question I am asking for first, this question applicable for all. So here it is A cell. Here you consider it is B cell. Here you consider it is C cell. So in A cell, number of chromosome, number of chromatid, number of DNA, and amount of DNA I am writing here equal to 4C. Now you answer everything for all question in sequence, so we will save our time. You tell me in A cell how many chromosome is there? You answer it. I will not answer. A cell how many is there? A cell how many chromosome is there? Say number of chromosome in A cell fast. 1. Number of chromosome is equal to number of centromere. Every time I told. So I am making centromere everywhere. Number of chromosome is always equal to number of centromere. This green color dot is centromere everywhere. Okay, ji? How many number of chromosome is there? 4. How many number of chromatid is there? 1, 2, 3, 4, 5, 6, 7, 8. Every line is a chromatid. So number of chromatid is 8. Number of DNA is 8 because I told few minutes before number of chromatid and number of DNA is always equal. An amount of DNA I have already given 4C. Now can we answer it for second question? And can we answer it for second question? B. So number of chromosome in B, I am writing in sequence. 2. Number of chromatid is 4. Number of DNA is 4. Amount of DNA is 2C. Can I say or not say? I am not writing, it will waste a lot of time. Now I am coming for C. Number of chromosome 2. Number of chromatid 2. Number of DNA, how many? 2. And amount of DNA C. Okay. So I am telling you one thing. If you assume C equal to 5 picogram, it is gamete. After meiosis II, who is forming? Gamete, that is egg or a sperm cell. So many time in paper we ask question, in egg cell, gamete cell, egg cell, sperm cell, if assume the amount of DNA is 4C, sorry, amount of DNA is C, then what will be the amount of DNA present in the cell, either in which phase? After S, G2 or metaphase, getting the things, metaphase 1, getting the things, or metaphase. So here it is which one? 4C. So after meiosis, you see now in daughter cell, here 4C, here 2C, here C. Amount of DNA become 1/4. After meiosis, amount of DNA become 1/4. So 5 picogram converted into 20 picogram. And suppose that anybody asking, ki in your, I am removing your skin cell and at G2 stage, and here it is 80 picogram of DNA is there. Assumption. Here at G2 stage, I am finding 80 picogram of DNA. Then in human sperm cell, how many picogram will present? Here it is 80. So sperm will, here it is 4C. A sperm becomes C. So 80 picogram, 80 into 1/4. How much is it? Are you getting the idea? Understanding the idea? Clarity came in the mind or not? Now I am considering it 2n. It become n. It is also n because number of chromosome 4, 2. So meiosis I is reductional division. And meiosis II n remain n, it is equational division. During meiosis I, who divide? Homologous chromosome divide. Homologous chromosome divide. And during meiosis II, who divide? Sister chromatid divide. Understand? Can we move on the next chapter? Okay, ji. Done. Otherwise, there is nothing a limitation of anything. Okay. So now we are going to talk about cell structure chapter, and there definitely we will take the help of NCERT and moving it faster. So let's come. Writing is taking more time now, but satisfaction came with writing. Okay, anyhow. See, in this chapter, what you have not to forget is cell is the smallest or basic unit of life. Every living organism are compulsorily made up of cell. Got the idea? I will tell few important lines. Beside of I will say, in marathon, you forget the purpose of the marathon. In marathon classes, we not came to teach you something new, not came to clear your concept, simply came to talk the hits area. Either you know or not know. If you know, well and good. If you don't know, it will get a revision for you. That is the only purpose. Got the idea? It is not a purpose to teach, to explain. And see now, we face maximum difficulty in dealing principle of inheritance, because it is highly conceptual chapter. Nothing a theory is there to explain. So what to explain? And if you go for explanation, entire time will waste in that only. That's the reason I say namaste to that chapter and move ahead. Look, give your attention. Just give me a break of 30 seconds, 1 minute, continuously speaking, sometime 30 seconds to 1 minute. Then I'm going to finish this chapter. Don't worry. Haji. So let's come on this chapter. It is also very important chapter for question asking. So don't underestimate it. Very important chapter. After that, we will go for another important chapter, that is plant growth. PGR, question going to ask. Look. So we are coming on cell. What I told, cell is the smallest unit of life, basic unit of life. Anything smaller than cell have performing living process. Got the idea? So any kind of cell have one property. They perform independent existence and they perform essential function of the life. First living cells, who describe it? Leeuwenhoek. Cell theory given by Sleeden and Swan. Sleeden was a botanist who told plants are made up of cells. Swan was a zoologist. Swan told in animal cell outermost covering is thin and thin membrane, and presence of plant cell is a characteristic feature of, presence of cell wall is a characteristic feature of plant cell. That is told by Swan. Ki plant cell have cell wall. Unique feature of the plant cell is what? Cell wall. Later on, Rudolf Virchow add one line in the cell theory, that is omnicellula, ecellula, and he also added all cell arise from pre-existing cell. Got the idea? In prokaryotic cell, you don't forget about the function of mesosome. In prokaryotic cell, mesosome is there. That is the extension of extension of which one? Plasma membrane. And these mesosome perform a variety of role. What role mesosome performing? Don't forget. Mesosome is responsible for which function? That is a very important line, key point. Mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicle, tubules, and lamella, and they help in cell wall formation, DNA replication, its distribution to the daughter cell. It also helps in respiration and secretion process, and it increases the surface area of the plasma membrane for enzymatic content. Increasing the surface area, cellular respiration, secretion, cell wall formation, DNA replication, and distribution to the daughter cell. In a prokaryotic cell, these all jobs done by the mesosome. In some photosynthetic cell, prokaryotic cell, chromatophores is also present. That is also an extension of plasma membrane, and which contain pigment in it, but it present in the prokaryotic form. And you don't forget even about the plasmid here. Plasmid here. This is an important line. You don't forget this line. Plasmid is what? Extracircular genetic material. In bacteria, two type of DNA is there, prokaryotic cell. One is genomic DNA, main DNA. It is also circular DNA. Got the idea? And beside of that, there is an additional circular DNA present, that is smaller in size, that is plasmid, and these plasmid have a role in providing resistance to which one? Antibiotics. Got the idea? Resistance to antibiotics. Getting the thing. So it's changing the phenotype of the host cell. Now we are moving ahead. You must know about the which one? Flagella. Flagella have a role in locomotion. Fimbriae have a role in attachment. Pili have a role in formation of conjugation tube. Got the idea? These all are made up of protein. Flagella in bacteria made up of flagellin protein. Pili made up of pillin protein. Getting the things or not? Inclusion body is reserve material in the prokaryotic cell. Got the idea? They are reserve material in the prokaryotic cells. They are non-bounded by any membrane, not bounded by any membrane, and they lie freely in the cytoplasm. Example of inclusion body is phosphate granules, synophysin granules, glycogen granule, and gas vacuole. Got the idea? Gas vacuole is inclusion body present in blue, green, and purple and green photosynthetic bacteria, because when we hold the gas, it give a buoyancy to float these bacteria and get a proper amount of sunlight. Synophysin granule store starch. Starch is stored by synophysin granule. Got the idea? Now, eukaryotic cell. If we talk about the eukaryotic cell membrane have a quasi-fluid nature. This quasi-fluid nature is due to phospholipid bilayer in cell membrane. Don't forget this line. Most explained hypothesis, accepted hypothesis of cell membrane given by Singer and Nicholson. Got the idea? That is fluid mosaic model. Don't forget its name. The lipid and protein content in plasma membrane is variable. In human RBC, 52% is which one? Protein, and 40% is lipid. That data don't forget. And the most important one here is lateral movement is possible in the plasma membrane is due to phospholipid bilayer. So fluid nature of plasma membrane is due to phospholipid bilayer. And these fluid nature perform an important function, like fluid nature of membrane have role in growth, formation of intercellular junction, secretion, endocytosis and cell division. Now if we talk about transport across the plasma membrane, when this transport occurs without expenditure of energy, it is called passive transport. Example of passive transport is which one? Diffusion. Diffusion, either simple diffusion or facilitated diffusion. Transport of water is called osmosis. So osmosis is a special type of diffusion in which water is moving across the plasma membrane. Got the idea? Is it clear? Now when transport of substances occur through plasma membrane with the help of expenditure of energy, ATP, it is called active transport. So passive transport occur from high to low concentration, it is toward the gradient, and active transport occur from low to high concentration, it is against the gradient. Example of active transport is sodium potassium pump. Got the idea? Sodium potassium pump. Now we are coming on cell wall. So if we talk about composition of algal cell wall, you keep in mind cellulose, galactans, mannans and mineral like calcium carbonate is present in algal cell wall. In higher plant, cellulose, hemicellulose, pectin and protein. Primary cell wall is present either in meristematic cell or in permanent cell. It is a universally present cell wall in every kind of cell, either meristematic or permanent cell. When cell when cell get mature, formation of secondary cell wall will occur, and which occur toward inner side of the primary cell wall. Got the idea? Middle lamella is a cementing material between two plant cell. It acts as a glue and hold the two different neighboring cell together. It is made up of mainly calcium pictate. And don't forget about the plasmodesmata. Right? What is plasmodesmata? Cell wall and middle lamella may be traversed by plasmodesmata, which connect the cytoplasm of neighboring cell. So from one cell to another cell, transport of material occur with the help of plasmodesmata. Got the idea? Who is going to transverse by cell wall, and middle lamella is going to transverse by plasmodesmata. Got the idea? Now we are moving ahead. Endomembranous system. How many cell organelles form the endomembranous system? Mitochondria, chloroplast and peroxisome is not a part of endomembranous system. Not a part of endomembranous system. Who is a part of endomembranous system? Endoplasmic reticulum, Golgi complex, lysosome and vacuole. Why they are called endomembranous system? Because their function are coordinated to each other. Function of ER, Golgi complex, lysosome and vacuoles are interconnected to each other. Right? Now we are coming on which one? Endoplasmic reticulum. What you don't forget from ER is, it is a which kind of a structure? Network or what happening? It is a network or reticulum of tubular structure present in the cytoplasm. It divides the cytoplasm into two parts. One is intraluminal and another is extraluminal. Extraluminal is cytoplasm. Two type of ER is there. When ER getting ribosome over it, it is called RER. When not get ribosome over it is called ACR. RER present near the nucleus, ACR present toward the plasma membrane. These RER present abundantly in protein synthesizing cell, and ACR is present abundantly in lipid and steroidal hormone synthesizing cell. Don't confuse. ER don't form protein. Protein is formed by ribosome present on the ER. So lipid synthesis and steroidal hormone synthesis, who is associated? A smooth endoplasmic reticulum, and ribosome on rough ER forming which one? Forming which one? Protein synthesis. RER associated with protein synthesis, ACR associated with lipid synthesis. Now, Golgi complex, discovered by Camillo Golgi. On honor of this, we have given the name Golgi complex. It is present as densely stained reticular structure near the nucleus. It consists of many flat disc-shaped structure called cisternae. Diameter is 0.5 to 1 micrometer. Got the idea? So what are the functions? These are stacked parallel to each other. These cisternae are present parallel to each other like this, stacked over each other. Is it clear in the mind? Now, it is a cell organelle with polarity. Golgi complex toward nucleus is called cis, and toward plasma membrane is called trans. Cis is also called forming phase. Trans is also called maturing phase. Got the idea? Cis phase is convex phase and trans phase is concave phase. So this is the cell organelle with polarity. Are you getting the idea? You're getting the idea? What are the function of the Golgi complex? Golgi complex is associated with packaging of material. Where it is written? Anywhere written? Packaging of material. Packaging of material. What the idea? It to be delivered either to intracellular target or secreted out of the cell. So suppose that any macromolecule transported either inside the cell or out of the cell, it is done by which one? Golgi complex. So packaging, secretion, and even glycosidation or glycosylation, formation of glycoprotein and glycolipid also done by which one? Golgi body. Any protein from ER came at cis phase of the Golgi complex through a vesicle, and they are contacted. These protein moving toward trans phase. When during this movement from cis to trans phase, chemical modification occur, that is the glycosylation. During that time, who will form? Glycoprotein and glycolipid will form by the Golgi complex. Lysosome formed by Golgi complex. Many vesicle of the Golgi complex fuse together and making a bigger vesicle, that is called lysosome. What the idea? It contains more than 50 different hydrolytic enzymes, that is called acid hydrolysase, and they are working best at acidic pH around 5 pH. Getting the things or not? The enzyme present here is what? Lipase, protease, carbohydrates, etc. They are at acidic pH, optimally active at acidic pH. Got the idea? They are digestion of what? Carbohydrate, protein, lipid, and nucleic acid. So these lysosome associated with intracellular digestion, and it is also called suicidal bag of the cell. Now we are coming on vacuoles. Got the idea? If you talk about vacuoles, it occupies 90% of the area. 90% of the area of volume of the cell. It is covered by a membranous structure called tonoplast. These large central vacuole have higher concentration than cytoplasm. Getting the things or not? Is it clear? Right? What vacuole is doing? It contains water, sap, excretory product, and other material not useful for the cell. Contractile vacuole is responsible for osmoregulation in amoeba-like organism, and food vacuole formed by formation or engulfing of food particle. Now, mitochondria and chloroplast, these two are semi-autonomous organelle. Everybody know about it. Plastid, you don't forget here. Mitochondria, nothing like plastid. You don't forget about leucoplast. So leucoplast is a colorless plastid. It is a storage in function. It is colorless plastid. It is a storage in function. Amyloplast store carbohydrate. Example in potato. Elioplast store oil and fat, whereas aleuroplast store protein. Got the idea? Otherwise, you know a lot of about which one? This one, ribosome, no need. Simply I am telling, sometime not in NCERT, but going to ask a question. Two ribosomal subunit hold together by which one? Magnesium ion. Not in NCERT. Cytoskeleton. Cytoskeleton is what basically network of filamentous proteinaceous structure consisting of microtubule, microfilament and intermediate filament. Right. These all together called what? Cytoskeleton. These cytoskeleton involve in which function? Many function. What are the many function? Mechanical support, motility, maintenance of shape of the cell. Last year PYQ. Last year this question came, and what were the option given? Motility. So what the job is done by the cytoskeleton? Support, motility and maintenance of cell. It is consist of microtubule, microfilament and intermediate filament. Got the idea? Now next, cilia and flagella arise from, it have 9+2 arrangement. Another thing arise from centriole like structure, basal body. So cilia and flagella have two components. One is plasma membrane, or it is also called periplasmic membrane. Periplasmic membrane. Another is axoneme. Axoneme have 9+2 arrangement. 9 peripheral doublet and 2 central singlet. Got the idea? So this is about the cilia and flagella. Keep in mind, peripheral doublet, central singlet. Axoneme is what? Have 9+2 arrangement. Centrosome and centriole. Many times a student confused. So this is 2 centriole present perpendicular to each other. They are surrounded by a amorphous layer. This is not the membrane, and this structure is called centrosome. So centrosome is present in animal and also in some lower plant, but it is absent in higher plant. If you go for a transverse sector of this centriole, these two are centriole, you got 9+0 arrangement. 9 peripheral triplet is there. Peripheral triplet are there. Peripheral triplet means 3 microtubules in one triplet. And it have a which kind of a structure? Cartwheel type of structure. Got the idea? So this is centrosome and centriole. You will find somehow in center there is a hub is there and 9 peripheral triplet are there. It is like this. I'm not, how many? 1, 2, 3, 4, 5, 6, 7. And it is connecting like this. AC linker, AC linker, AC linker. In hurry, what's happened? Not with this. AC link. Oh. With AC, you think like this. It is going to attach like this. And from here, it is going to attach with the hub. Not the idea. I'm not drawing the detailed structure. It will take a lot of time. So I think NCERT will not have diagram of it. So it have a cartwheel like a structure. You keep in mind. Cartwheel like a structure. So now we are going to talk about nucleus. What you don't forget from nucleus is described by Robert Brown. First thing, nucleus stained by a basic dye. Second thing, and the name who stain material inside the nucleus is stained by basic dye and given the name chromatin by Fleming. Got the idea? So if you talk about nucleus, it is surrounded by nuclear membrane. Double membrane are there which are parallel to each other, but this membrane is not continuous, so nuclear pore is there. A space between the nuclear membrane is how much? 10 to 50 nanometer. It is called perinuclear membrane. A space, got the idea? Now, through nuclear pore, who is passing from one direction to another direction? Both RNA and protein passing from one direction to another direction. Got the idea? So, inside the nucleus, what you are finding? The nuclear matrix or nucleoplasm contain, inside the nucleoplasm, who is there? Nucleolus is there and chromatin is there. Nucleolus are a spherical structure present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm. Which nucleolus not bounded by any membrane, and it is very important line. It is a site for active ribosomal RNA synthesis. Who is the site for this? Nucleolus. So examiner will ask active synthesis of ribosomal RNA occur on which one? Nucleolus. Get the idea? Is it clear in the mind? Now if we talk more, chromatin condense and forming which one? Chromosome. So where two sister chromatids join, that area is called centromere. I am just writing here. Here it is what? Centromere. What happening? Okay. And inside of the centromere, kinetochore is there. You try to understand here. This is one chromatid, it is another chromatid join here. It is centromere, and this side kinetochore is there. And with kinetochore, who is attaching? A spindle fiber is attaching. Now, if we talk about something more. So, based on the position of chromosome, there are four type of chromosome is there. Centromere in middle, metacentric, it is V shape. Away from middle, submetacentric L shape. Near to tip, it is J shape, acrocentric, and on tip, it is I shape. It is telocentric. It is not there. Diagram is somehow, both are looking of acrocentric. It must be your more tipper. Got the idea? So in chromosome, you just see, it is a chromosome. In a chromosome, I am asking here a question. How many centromere is there in this chromosome? One centromere. How many kinetochore? Two kinetochore. How many chromatid? Two chromatid. How many DNA? Two DNA at metaphasic chromosome. And satellite may present or not present. Satellite is the bulge out region after the secondary constriction. It is secondary constriction. It is not compulsorily present in every chromosome. Got the idea? How many telomere is there? 1 telomere, 2 telomere, 3 telomere, 4 telomere. Examiner can ask this type of question. One chromosome at dyad stage. It is dyad of the chromosome. Have two chromatid, 4 telomere, 1 centromere, 2 kinetochore, 2 DNA, 4. Got the idea? These are the things you keep in the mind. So this is over. Now another important chapter, and especially we will become somehow more slow there, because also two questions probably we are going to deal from here. In fifth, I hope two questions must came from which one? Plant growth. Theoretical chapter. Deliberately I am talking right now on theoretical chapter, because in conceptual chapter it required teaching. And you can't understand concept in few minutes or in a hurry. So that's the reason I am just not talking much on conceptual chapter. I am just talking on which one? Factual chapter more. And this is one of the chapter which have a very high weightage for the examination. Got the idea? So now we are going to talk about PGR. And especially in PGR, we will start our story with the ethylene. coolly we will talk about. Not in hurry. Here we are not in hurry because compulsorily question will come. So if 2 questions came from PGR, it is a matter of 10 marks. 4, 4, 8, but you will miss, then it is 10 na. So it is a matter of 10 marks. So don't become impatient. It is most important things. You are listening, we are speaking. So doing more hectic job than you. So listen it carefully. We are starting with which PGR? Ethylene. What to not forget from here, I am telling you. Now give your attention about the ethylene. First thing, it is a gaseous PGR. It acts as both promoter or inhibitor. But most of the work of the ethylene is like an inhibitor. So we place this along inhibitor, but it has both promoter function and inhibitor function. Got the idea? Which part of the plant synthesizing more amount of ethylene? So it is synthesized in large amount by tissue undergoing in senescence and ripening fruit. Tissue who undergoes in senescence, I am telling very coolly and calmly about the ethylene because it is very important. PGR is very important. At least 2 questions will come in NEET. 2 questions is the game of 2 questions. So what I am saying, it is synthesized in large amount by tissue undergoing in senescence and ripening fruit. So what it is doing, it is performing triple response. What is this? First, influences of ethylene on plant include horizontal growth of seedling, don't forget. Second, swelling of axis. Third, apical hook formation in dicot seedling. What impact it is doing, ethylene influence it is doing on plant? Horizontal growth of seedling first. I am very slow right now. Swelling of axis. Third, apical hook formation in dicot seedling. Second, ethylene promotes senescence and abscission of plant organ, especially leaf and flower. Senescence means getting older, abscission means falling. So ethylene promotes senescence and abscission of plant organ, especially which part? Leaves and flower. Don't forget this line. Ethylene is highly effective in fruit ripening. Okay, see, it enhances respiration rate during ripening of the fruit. And this rise of rate is called respiratory climacteric. So which PGR is performing respiratory climacteric? Ethylene. Done. Now what another work done by the ethylene? What another work done by the ethylene? It breaks seed and bud dormancy. Breaking the seed and bud dormancy. Don't forget this. It initiates germination in peanut seed. Germination initiated, right now, a sprouting of potato tuber also performed by ethylene. Ethylene promote rapid internode or petiole elongation in deep water rice plant. Your state is a rice growing belt, so you grafting the rice, new rice plant in the water. So due to ethylene, it grow very faster in the water, so it not sink in the water. So it help leaves or upper part of the shoot to remain above the water. Ethylene also promote root growth and root hair formation. Root growth, root hair formation, breaking of bud dormancy, germination of seed, a sprouting of potato tuber, promote rapid internode elongation. These are which kind of job? Inhibitory job or promoter job? These are promoter job. Got the idea? Ethylene also none, thus helping plant to increase their absorption area. Ethylene used to initiate flowering and synchronization of fruit set in pineapple. Don't forget. Flowering also induced in pineapple by auxin, but it is flowering also and synchronizing the fruit cell also. It also induced flowering in mango also. Got the idea? So it is regulate many physiological process, many function we have seen in ethylene. No one have that much function. That's the reason I am starting from here. It is one of the most widely used PGR in agriculture practice. The most widely used of the ethylene as which one? Ethophone. Commercially in market, ethylene is available as ethophone. In aqueous solution, it is readily absorbed by plant and released slowly. What the idea? Ethophane hasten fruit ripening. Hasten means fasten. In tomato example, apple, and accelerate abscission. In flower and fruit, thinning of cotton, cherry, walnut is done by which one? Ethylene. It produces femaleness in cucumber, so induce the productivity. So these are about the ethylene PGR. There is a maximum chances one question going to ask from here in the NEET exam. Got the idea? Ethophone is available in market commercially. People use it for enhancing the ripening of the fruit. It readily absorbed by the plant and it is releasing slowly and responsible for ripening of the fruit. That is ethophone. Got the idea? And many function, root growth. It is written, na? I am telling here, na? Which one? Where root word was there? Ethylene also promote root growth and root hair formation. What the idea? These are also the job of the ethylene. Don't forget it. So ethylene is over. After ethylene, we are talking about the oxygen. I am also dealing it with the help of priority. Second PGR, oxygen, I am going to talk. So what you not forget about the oxygen, let the NCERT, leave the NCERT, I am telling you. Oxygen. Oxygen, what it is doing, the main function. It delays senescence and abscission in young leaves or young plant. A young leaf or young plant, it is delaying the abscission or senescence. In old part of the plant, it is promoting. One function. Oxygen is responsible for formation of root hair at the stem cut. It have used in grafting. So when you spray the oxygen at the stem cut part, root hair will come from there. Root will arise from there. Rooting at stem cut. It is called rooting at stem cut. Next, oxygen is doing apical dominance. Plant grow more in the height. It is due to oxygen. Apical dominance. Many times students confuse oxygen not used in tea plantation. Oxygen is a problem for tree plantation because tea plant become too tall. How you can pluck the leaf of the tea? So NCERT wrote, in tea plantation we are doing decapitation, cutting the tip, and when we are cutting the tip, who will not release? Oxygen will not release, branches will grow more. Able to understand or not? So we are cutting the tip to avoid the plant to grow the height more. So plant remains somehow dwarf, so we can easily pluck the leaf from there. So decapitation means cutting the tip. Even in hedge making, we are cutting the tip. Pruning we are doing. So in tea plantation and which one? Hedge making, there is no need of oxygen. So we are cutting the tip, so oxygen not produce, so plant not grow much in the height. Are you getting the idea? Is it clear? Is it clear? Another function of the oxygens is what? Basically flowering in pineapple. Another function of the oxygen is xylem differentiation and cell division. Another function of oxygen is parthenocarpy in tomato. Getting the idea or not? Parthenocarpy in tomato. Is it clear? Done. Parthenocarpy is what? Seedless food for fruit formation. Do you know the difference between parthenogenesis, parthenocarpy and apomixis? Parthenocarpy, I am telling because this term came. Parthenocarpy, parthenogenesis and apomixis. All three events occur without fertilization. All three events occur without fertilization. First thing. Parthenocarpy, formation of fruit without fertilization is called parthenocarpy. Fruit becomes seedless. Fruit become seedless. Parthenocarpic fruit become seedless. Second, parthenogenesis, formation of embryo or organism without fertilization. Like in honeybee, male honeybee, formation of embryo or organism without fertilization is called parthenogenesis. And apomixis, formation of seed without fertilization. Formation of seed without fertilization is called apomixis. So these are the three related terms where students sometimes confuse. All the three events occur without fertilization. Clear? Now we are moving ahead. Now 2,4-D used as which one? weedicide. It kill the dicot weeds when you are growing in the monocot lawn. Suppose that you have a beautiful bungalow, grass is there, and some unwanted plant grown. It not look beautiful, na? So you are spraying which one? Oxygen to kill the dicot weeds. Got the idea? So these are about the important functions of oxygen. Don't forget it. Now we are coming on which one? Gibberellic acid. What is the important function of the gibberellic acid? Most common GA is what? GA3. And most studied GA is what also? GA3. Gibberellic acid 3. More than 100 type of gibberellic acid are isolated from different sources. Getting the things or not? Now we are going to talk about the physiological function of the GA. Physiological function of GA. Name is saying, no need to mug up. All GA are acidic. Gibberellic acid. Got the idea? So what their ability to cause an increase in length of axis used to increase the length of grape stalk. So we are increasing the stalk of the grape. So size of the grape and productivity of the grape will increase with the help of gibberellic acid. Gibberellic acid also improve shape and size of the apple. Elongate the apple and improving the size of the apple also. It delay the senescence because it is a promoter. Got the idea? It also help the fruit to kept in the plant for the longer time. So market period get extended. Suppose that you have 10 acre of mango farming. And right now mango price is not good. You are spraying gibberellin. These mango plant fruit will remain in plant for a longer time. You can sell it when you find the better price is coming. Got the idea? It speed up the malting process which used in brewing industry. Got the idea? It increased the productivity of sugarcane by 20 ton per acre because it increased the gibberellin have that power. You spray gibberellin in dwarf plant become tall. God made something like human also na be spray and become tall like, but nothing is there. Sorry to say, but in plant it is there. You spray the gibberellic acid, short plant become tall. Nothing for us. Okay, right? So spraying the gibberellin, what happening? Spraying juvenile conifers with gibberellic acid hasten the maturity period. Hasten means fasten. This leads to early seed production. These also promote bolting, that is internode elongation, just prior to flowering in beet, cabbage, and many plants with rosette habits. Right. So it have role in which one? Increasing the productivity of apple, grape, sugarcane. Getting the idea or not? But NCERT wrote directly with the sugarcane. Right. Okay. So now we are moving ahead about the another PGR. That is cytokinin. What you don't forget about cytokinin is, what? Better you just see everything here, because we are not in position to leave anything from this chapter. It is very important for exam. So cytokinesis have a specific effect on cytokinesis. Right, and discovered as kainate in a modified form of adenine, a purine, from autoclave herring sperm DNA. Kainate isolated from animal source. Got the idea? Right? So cytokinin-like substances isolated from plant source is what? G18. Kinetin from animal cells, G18 from plant source. It is from where? Corn, kernel, coconut milk, etc. Is it clear? Several naturally occurring cytokinins and some synthetic compound with cell division promoting activity. They are promoting which one? Cell division. Natural cytokinins are synthesized in region of rapid cell division occur. Which area it is producing where rapid cell division occur? What the idea? Example is root apices, developing shoot bud, young fruit, they are releasing which one? Cytokinin. Now I am coming on the most important area, the function. What function cytokinin is performing? So cytokinin is help in production of new leaves, chloroplast in leaf and lateral shoot growth and adventitious shoot formation. Once again listen it. Cytokinin function, production of new leaf, production of chloroplast in leaf, production of lateral shoot growth, and it is responsible for adventitious shoot formation. Cytokinin also help to overcome apical dominance. It is antagonist of auxin in this function. Antagonin want plant grow in height. Cytokinin want plant grow in branches. So when you want a plant to grow tall, cut the branches. And when you want to plant to make more branches, cut the tip. Horticulturists do it. Got the idea? In timber yielding plant, you want more height. So cut the branches. In mango, you want more branches. So cut the tip. So what happened? Branches will come more. Is it clear in the mind or not? Now we are coming on which one? Abscisic acid. The last PGR. In this chapter, we will not deal anything other than PGR. Abscisic acid. It is a true sense growth inhibitor. Got the idea? Role in regulating abscission and dormancy. Right. It is stimulating the closure of the stomata and increase the tolerance of plant to various kind of stress. It is the stress hormone of plant. What doing by in your body by adrenaline, same work done by plant by through which one? ABA. ABA play important role in seed development because it not allow the seed to germinate, to break the dormancy. Getting the things or not? So maturation and dormancy. It promote the dormancy. Got the idea? So seed development occur. ABA help to withstand desiccation and other factor unfavorable for growth. Unless bad condition is there, they not allow the seed to germinate. Otherwise, what happen? New seedling will die. It is antagonist of GA, opposite function of GA. Got the idea? So PGR is over. This chapter is also done for examination. One thing is there. I am asking from you. Chemical name and discovery. It is also important. Auxin is what? Chemically indole acetic acid. Cytokinin is adenine derivative, N6-furfuryl aminopurine. Who derive from terpene? I am asking to you. Who derive from terpene? Derivative of terpene. Which PGR? GA. Abscisic acid is which one? Carotenoid derivative. So ethylene is a gaseous PGR. Don't forget it. Now experiment. Scientist associated with discovery of gibberellic acid. Kurosawa. Got the idea? Scientist associated with discovery of auxin. F.W. Wendt. Got the idea? Darwin and his son perform experiment with unilateral light and bending of the coleoptile. Getting the idea? Darwin did which one? Bioassay of auxin is also performed by which one? F.W. Wendt. They work on which one? Oat seedling. Right now, Avena curvature test. Okay, ji? Right? Now, if we talk about cytokinins, scientist associated with that is what? In the discovery, Skoog and Miller. Skoog and Miller. Miller performed the crystallization. Ethylene is discovered by which one? Cousins. They find like what? From ripen orange, some volatile chemical is releasing which enhancing the ripening of banana. And 3 independent researchers developed which one? Abscisic acid. And they have given the name as what? Inhibin B, dormine and abscessin. And they all together called abscisic acid. So this is about the discovery. Now we are coming on next chapter. You tell me which chapter in your priority. Cellular respiration should I teach? ATP calculation and all you want? In 15 minutes I will fix it. Entire cellular respiration. You wish? I am asking your priority index. Okay, cellular respiration. So no need of book. I am starting now. Cellular respiration. Look here. Glucose converting into pyruvic acid. Let me write in left side so I will find a lot of space. Otherwise it's not a scroll, na. Glucose converting into 2 molecules of pyruvic acid. And this event is called glycolysis. During glycolysis, a 2-step ATP needed. At 1 step, 2 NADH produced. And at 2 step, 2 ATP produced. These ATP produced by SLP, substrate level phosphorylation, direct release of ATP. So this is the outcome of glycolysis. Now this 2-pyruvic acid undergoes in mitochondria and undergoes in link reaction. I am telling aerobic respiration first, link reaction. And it converted into 2 molecules of acetyl coenzyme. When it is converting into 2 molecules of acetyl coenzyme, 2 NADH produce. Now this 2 acetyl coenzyme undergoes in Krebs cycle. So in Krebs cycle what happening? Oxaloacetic acid converting into citric acid. First stable product, hence it is called citric acid cycle, or it is a tricarboxylic acid, that is called TCA cycle. Got the idea? Then it is converting into alpha ketoglutarate acid. Then it is converting into succinyl coenzyme. Then it is converting into succinic acid. Then it is converted into which one? Malic acid. Then converting into oxaloacetic acid. I am writing few of the steps. So what happened in this step? When citric acid converting into alpha ketoglutarate acid, oxidation occur and formation of NADH2 occur. When alpha ketoglutarate acid converting into succinyl coenzyme, oxidation occur and NADH2 formation occur. And when succinyl coenzyme A converting into succinic acid, I wrote more step than NCERT. Here GTP or ATP produced by SLP, substrate level phosphorylation occur. When succinic acid converting into malic acid, NADH formation occur. And when malic acid convert into oxaloacetic acid, NADH formation occur. And because 2 acetyl coenzyme, so 2 Krebs cycle is there. So everyone who is forming, you multiply by how many? 2. Because 2, I am simply multiplying everywhere with 2. Nothing I did more here, 2. Now I am asking a question to you. People, कि when one glucose undergoes in complete oxidation, you count and tell me how many total NADH produce. You count and tell me how many total NADH produce. Two NADH produce here. Let me change the color. 2 NADH produce here. 2 NADH produce in link reaction, 2 NADH in glycolysis. 6 NADH produce in which one? During Krebs cycle. So total how many NADH? 6 in Krebs cycle, from 2 Krebs cycle, 2 from 2 link reaction, 2 from glycolysis. Total from NADH from glycolysis. NADH I am writing. From glycolysis how many? 2. From link reaction how many? 2. From Krebs cycle how many? Link reaction how many copy occur? I am also writing 2 copy of link reaction, 2 copy of Krebs cycle, 1 copy of glycolysis. So from 2 Krebs cycle how many it came? 6. 6 plus 4 equal to 10 NADH. 6 plus 4 equal to 10 NADH. How many FADH produce? 2 FADH. Got the idea? 2 FADH2 produce from 2 Krebs cycle. Got the idea? So how many direct ATP produce? By SLP 2 to 4 plus 2 6, and I am doing minus 2 equal to 4. Why I am doing minus 2? Because 2 ATP consume in the glycolysis. Got the idea? So direct ATP by SLP 6 minus 2 equal to 4. Now total ATP how came? How total ATP came? 10 NADH give 10 into 3 ATP equal to 30 ATP through ETS. Because 1 NADH give how many ATP? 1 NADH give 3 ATP. 2 FADH to give 2 into 2 equal to 4 ATP through ETS. Total how much ATP? 34 ATP through ETS or oxidative phosphorylation. Are you getting the idea? Through ETS or oxidative phosphorylation, and 4 from SLP. So 34 plus 4 equal to how much ATP? 38 ATP. So this is the justification of 38 ATP. Got the idea? This is the justification of 38 ATP. Did you got the idea? Clarity came. Net gain of ATP is how much? 38 ATP by 1 mole of glucose. Now I am telling you ETS. You want ETS? In 2 minutes you want ETS, 3-4 minutes? Maximum what the time will require to draw? I am telling ETS. Unnecessarily you people make a nightmare for ETS. Look. What the time required to draw the diagram, it is, I am not using NCERT diagram because you will unnecessarily confuse. I am telling you very easy diagram. Here it is outer membrane, perimitochondrial space, inner membrane. I am using the same thing but changing the sequence. Now look here, ETS. Here complex 1 is there. This is complex 2 is there. This is complex 3 is there. This is complex 4 is there. This is complex 5 is there. Done? Now you have to mug up in which complex what is present. So in complex 1 what is present? FMN, iron sulfur. And enzyme, what enzyme present along with complex 1? Tell me. NAD, tell me. Enzyme associated with complex 1. NADH dehydrogenase. Well done. NADH dehydrogenase. Complex. It can guide that. So here it is complex 1, complex 2, complex 3, complex 4, complex 5. Every complex name you have to keep in mind. Right. So I am writing first complex name is this. Second complex name is SD. Succinic dehydrogenase. Short name I wrote. Third complex name is cytochrome BC1 complex. Or it is also called cytochrome C reductase. C reductase. Complex 4 is which one? Cytochrome C oxidase. This name you keep in mind. Complex 5, ATP synthetase. ATP synthetase. You got the idea? Now give your attention. What present in complex 2? Tell me. In complex 2, who is present? FAD and iron sulfur. In complex C, who is there? Cytochrome B, iron sulfur, cytochrome C1. Do it by heart. Here copper A, cytochrome A, cytochrome A3, copper B. Here F node, CF node, CF1, all together called ATP synthetase. And in between two mobile carrier is there. One is coenzyme Q, UQ you can say ubiquinone. Another is cytochrome C. It is present toward outer side of the inner membrane. Now how the oxidation occur? Now in another maximum 2 minutes or 1 minute ETS will over. Now what happening? Look here. Give your attention. NADH undergoes in oxidation and converting into NAD plus. Electron from NADH moving like this. How it is moving you see. It is not going on complex 2. From 1 to ubiquinone going to complex 3. This is the route of travel. And finally electron received by half O2. So 2 electron transporting from NADH2 via different electron transport carrier finally received by which one? Half O2. This is the only job of the water. Half O2. Oxygen in the ETS. This is the only job of oxygen in your body. It acts as a terminal electron or hydrogen acceptor. They are taking 2 proton from matrix and converting into water. So oxygen reduced into water. So what a story happened, you just try to understand. NADH plus H plus converted into NAD plus, and here half O2 converted into water. Finally occur this. Who lose the electron, who undergoes in oxidation? NADH, who go in reduction? Half O2. It acts as a terminal acceptor of, right now. So what happening? Whenever electron is moving downhill in this potential energy scale, what happened? Release of energy occur due to electron movement. Electron when moving, it is like photosynthesis. Who is releasing? Energy is releasing. And this energy which is releasing used in pumping of proton. So they are pumping 10 proton. 4 proton at level complex level 1, 4 proton at complex level 3, and 2 complex at complex level 4. So what happening? What is the moral of a story? When NADH2 undergoes in oxidation, electron moving through different electron transport carrier. And here 2 mobile carrier is also there. From complex 1 to ubiquinone to complex 3 to mobile carrier cytochrome C2 complex 4 finally to oxygen half O2 taking which one? 2H from matrix proton and converted into water. In this electron movement, during these electron movement, energy release, these energy used in pumping of proton from where to where? Matrix to perimitochondrial space. Now there is a gradient creation occur, proton gradient creation occur. So what happened when proton gradient creation occur? Here it is inner membrane, here it is outer membrane, here it is inner membrane, here it is perimitochondrial space, here it is CF node, here it is CF1. CF node providing the facilitated diffusion of the proton, and when how many proton? All NCERT wrote 2 proton here, but in theory they wrote 4 proton. So when 4 proton return back from where to where? Perimitochondrial space to matrix, what happened? ADP plus inorganic phosphate join and make a ATP. So for 1 ATP production, how many proton is required here? 4 proton were moving down the gradient. So 10 proton give how much total? 2.5 ATP. In NCERT in lump sum it is written 3 ATP. When you go in your medical college, you will do every calculation with 2.5 ATP. Got the idea? But right now, you consider it is in lump sum. NCERT did a lot of nonsense there. They wrote here 2 proton, then 5 ATP came. So 4 proton they wrote in theory. 4 proton when returned back, they give lump sum 2.5. Right? Is it clear? Done? Over? 2.5 ATP NCERT wrote in lump sum 3 ATP. So this happened during the ETS when 1 NADH going for oxidation, it is going to produce how many ATP? How many ATP? 3 ATP. 1 FADH, if you talk about FADH, when it is going for oxidation, what happened? Look here. FADH2 converting into FAD, and it is moving like this. So it is pumping how many proton only? 6 proton. And when 6 proton pump, it is giving how many ATP? 1.5 ATP. In lump sum equivalent, it is written how many ATP in NCERT? 2 ATP. You got the idea? Is it clear? So FADH2 give 2 ATP in round figure, NADH2 give 3 ATP in round figure. Got the idea? Is it clear? Now you tell me what? Respiration, RQ, you know, getting the things or not? Now, which one? Morphology, anatomy, biological classification, what you want? Plant kingdom. Okay, ji. So first thing you don't forget in plant kingdom is here. Which area you just find difficult in plant kingdom, so I will help without NCERT, it become more faster. Which area you are stuck in more? In plant kingdom, which area you are stuck in more? Bryophyte and pteridophyte, okay. So I am saying you the bryophyta. Let me remove it. Coming on Android, it is better. Wait, speed will increase. Oh, what happened? So Bryophyta and Pteridophyta. Life cycle, I think you have problem, right now? Life cycle? Okay. So look, in life cycle, in any life cycle of the plant, you keep two things in mind. That basic I am telling you, it will help you. It is a cycle. It will help you to understand any life cycle. It is a sporophytic stage and it is gametophyte, assume. So gametophyte stage always, sporophyte stage always start from zygote, 2N stage. And first cell of the sporophytic stage is zygote. I am giving you a ray diagram of life cycle in 2 minutes first. First cell of the sporophytic stage is zygote. Zygote divide in by mitosis and forming a sporophytic plant body. A sporophytic plant body. These sporophytic plant body is forming which one? Have which one? A sporangium. A sporangium have what? A spore mother cell. A spore mother cell have always which ploidy? 2N. A spore mother cell have always which ploidy? 2N. So spore mother cell is last cell of a sporophytic stage and zygote is the first cell of a sporophytic stage. Keep it, it will help you understand that. Now they divide by meiosis. So meiosis is the event who convert a sporophytic into gametophyte and forming a structure N. N is a spore. A spore is the first cell of the gametophytic plant body. Now these spore divide by mitosis and forming gametophyte. Gametophyte, and these gametophyte is forming male gamete and female gamete. So gamete is formed by gametophyte and these male gamete and female gamete fuse, means fertilization, and it is forming zygote. It is forming zygote. So you can say gamete is the last cell of gametophyte and a spore is the first cell of which one? Gametophyte. Got the idea? So in plant, gamete always produced by which division? Mitosis. And in plant, a spore always formed by which division? Meiosis. You got the idea? Now you come on the bryophyte life cycle. I am just opening NCERT so it become more easy for you, and otherwise I am drawing itself, not a problem. And how much time you will open it, you can write as well. Look here. So bryophyta we are going to just discuss. Look here. Life cycle of bryophyta. So here it is gametophyte. Gametophytic plant body. So it is a gametophytic plant body. I am writing gametophyte up, not a problem, na? Gametophytic plant body it is. Anywhere you can write anyone. So it has sex organ. Gametophytic plant body have sex organ. Male reproductive organ is antheridium. And female reproductive organ is archegonium. Antheridium produce anthrozoids. Or you can say male gamete. Anthrozoids. It is anthrozoid. It is biflagellated anthrozoid. Archegonium is a flask structure in bryophyta. It is archegonium. And in center of it, it is egg cell is there. It is called neck. It is called venter. It is called stalk. Now what happened? These antheridium, anthrozoid, swim in water, enter into the neck and fuse with the egg and forming which one? Zygote. So this zygote I am writing this side to make you understand it is first cell of which one? 2n stage. It is n stage. It is 2n stage. Now this zygote divide by mitosis. And forming which one? Embryo. These embryo further forming which one? A sporophytic plant body. These sporophytic plant bodies not have independent existence. Who is more developed in bryophyta? In bryophyta, who is more developed? Gametophyte is more developed. Gametophyte is photosynthetic. I am drawing the structure of gametophyte here. It is rhizoid, main axis, and leaves. So it is gametophytic stage. It is diploid, sorry, it is which one? Haploid stage. A sporophytic stage is depend on gametophytic stage. So here I am drawing a sporophytic stage. It not have a independent existence. It is called capsule. It is called seta, and it is called foot. These all have 2n ploidy. It is sporophytic stage. It is gametophytic stage. So sporophytic stage is dependent on gametophytic stage in bryophyta. Now what happened? In capsule, spore mother cell is there. Spore mother cell. Spore mother cell. These spore mother cell divide by meiosis and forming haploid spore. It is haploid spore. Haploid spore. Now what happened to these spore? In these spore you will find that in liverwort directly germinate and form gametophyte. In liverwort. In mosses what happened? Indirect development occur. In moss first what happened? These are going to form which one? Protonema. And these protonema further going to forming which one? Bird. Protonema is a juvenile stage. And later on from bird, who is going to develop? Either gametophyte or secondary protonema. And from secondary protonema, gametophyte. So here indirect development occur in moss, direct development occur in liverwort. So this is the life cycle of which one? Bryophyta. If you talk about according to NCERT in bryophyta, which kind of life cycle is there? I am telling life cycle faster according to NCERT. So haplontic life cycle. If you talk about haplontic life cycle, example, most algae. What happened here? Gametophyte is free living, independent, photosynthetic. And a sporophyte is dependent on gametophyte. Got the idea? So now haplodiplotic. In haplodiplotic life cycle, where it occur? In two group it occur, bryophyta and pteridophyta. What happened in bryophyta? Let me tell you. In bryophyta, gametophyte is dominant and independent. A sporophyte is parasite or semi-parasite own gametophyte. In pteridophyta, very specific peculiarity is there. Both gametophyte and sporophyte have independent existence. Have independent existence. Got the idea? But who is dominant? A sporophyte is dominant. Now diplontic life cycle. Last. Diplontic. Gymnosperm and angiosperm. Angiosperm. Here what happened? A sporophyte is dominant. Is free living and dominant. And gametophyte is parasite over sporophyte. You got the idea? Is it clear? So this is about the life cycle. How much time we have? Wait a minute. So see, we are going to revise very fastly about morphology. Sweet potato is a modified what? Adventitious route for storage of food. Don't forget this line. Now, this is an area from where question is going to ask. Hypogynous superior ovary, where it is present, what is the example of the hypogynous condition? Mustard, China rose and brinjal, don't forget. Perigynous half inferior ovary, what is the example for perigynous? Palm, peach and rose. Epigynous is inferior ovary. Example is ray florets of sunflower, guava and cucumber. Got the idea? So these are the position of the ovary is important, keep in mind. Now another area which is highly important is estivation. Right? So valvate estivation present in calotropis, don't forget this line. Twisted estivation, it is present in which one? China rose, ladyfinger and cotton, don't forget this line. Imbricate estivation present in cassia and gulmohar, and waxillary estivation which have a papillinaceous corolla. 1+2+2 arrangement. Got the idea? This one is present in which family? Fabaceae family. P family, bean family. Got the idea? Is it clear? So this is about the estimation. Now, what are another area which is important from the flower is epipetalous condition. What's happening? Epipetalous condition present in member of family Solanaceae. Example is which one? Brinjal. Epiphyllous condition present in family Liliaceae. Epipetalous means what? When stamen fuse with which one? Corolla. Epiphyllous condition is what? When it fused with which one? Perianth. So this example is which one? Brinjal. This example is which one? Epiphyllous condition example is lily, family Liliaceae. Got the idea? Now, monoadelphous condition, when one bundle of the filaments are there, it is called monoadelphous. Example is China rose. Diadelphous, P family, P. Got the idea? And polyadelphous present in citrus. Also, don't forget this line. Now, from placentation, you have to keep in your mind. Also, match of the following question possibilities there. Marginal placentation. Wait, just a minute. This is marginal placentation. Examiner even asked to identify the diagram, but now it is diagram based question is not there. It is present in which one? P family. Got the idea? Marginal placentation. After that, we are going to talk about axile placentation. This is the diagram of axile placentation. Multilocular ovary is there, cross wall septi is there, ovule arise from the central axis. This type of placentations, if you find it present in China rose, tomato and lemon. Parietal placentation, ovule arise from the inner wall and it is unilocular but become bilocular due to formation of false septum. Such kind of placentation present in which one? Which family? Tell me. Family Brassicaceae, mustard family, it is parietal placentation. Now I am talking about the free central. It was multilocular but become unilocular and ovule arise from the central axis. Example of which one? Argemone. No, margemone, dianthus and primrose. Mustard and argemone is the example of parietal placentation. Dianthus and primrose is example of free central placentation. Lastly, basal placentation. Basal placentation present in the sunflower family, marigold. Getting the thing, single ovule attached at the base, and even basal placentation present in family Poaceae. Family Poaceae. So in family Poaceae and family Asteraceae, which kind of placentation is there? Basal placentation is there. Got the idea? Now nothing in fruit, seed, we are coming on floral formula in the family. Only one is given in this. Solanaceae. So if we talk about floral formula, only given in NCERT is which one? Solanaceae. So what you are finding in Solanaceae? It is actinomorphic, bisexual, calyx, 5 fused sepal are there, corolla, 5 fused petal is there, stamen 5 and free is there, but it fused with which one? Corolla, epipetalous condition, gynoecium bicarpellary. Got the idea? Now if we talk about floral formula of Fabaceae, how to write the floral formula of Fabaceae? It is zygomorphic, it is bisexual, it is K5, it is C1 plus 2 plus 2, it is A9 plus 1, and it is G1 superior ovary. This is the floral formula of which one? Leguminaceae. Leguminaceae, legume plant. Got the idea? This is the floral formula of Leguminaceae. Liliaceae, what is the floral formula? Bracteate, right? Bisexual, perianth, 3+3 fuse, stamen, 3+3, and it is fused which one? Perianth. Then you can say about gynosium, hypogynous, tricarpellary fuse. This is the floral formula of which one? Liliaceae. What is the idea? Mainly focus on NCERT material now. They have given additional families, but I doubt either they ask or not ask. I'm not very confident about it. So these are the important area from floral formula. Keep in mind now Brassicaceae is also there. Tell me the floral formula of Brassicaceae. It is actinomorphic, bisexual, K2+2, C4. After that you can say about which one? A2+6 and gynosium superior ovary bicarpellary fuse. Right? So this is about, I hope now you are tired and even I am tired, time up. So remaining you do by yourself. Bye-bye. Thank you very much for your patience for listening. Bye. Anatomy, do yourself. Bye.